Assalamualaikum warahmatullahi wabarakatuh. Tabi pun. Alhamdulillahirabbil alamin. Assalatu wassalamu ala asrofil anbiya'i wal mursalin wa ala alihi washabihi ajmain. Amma ba'du. First of all, let us praise our God, Allah Subhanahu wa Ta'ala, the most gracious, the creator of the universe. And peace be upon Prophet Muhammad Sallallahu Alaihi Wasallam as the last messengers of Allah Subhanahu wa Ta'ala.
Pertama-tama marilah kita panjatkan puji dan syukur kepada Allah Subhanahu wa Ta'ala yang Maha Pemurah lagi Maha Penyayang pencipta alam semesta ini. Salawat serta salam kami sampaikan kepada junjungan Nabi besar kita Nabi Muhammad Sallallahu Alaihi Wasallam.
Our Honorable Director of University of Lampung, our Honorable the Head of Center for Research and Development of Medicinal Plants and Traditional Medicines, Bapak Ahmad Saiku MSc, our Honorable the Head of Institute for Research and Community Service, Dr. Lusmelia Afriani, our Honorable all of the Dean of University of Lampung, our Honorable all the plenary speakers, our Honorable the Chairwoman of the event, Ibu Endang Linirin Widyastuti PhD, our Honorable all the participants and presenters, and our beloved guests.
Yang terhormat Rektor Universitas Lampung, yang terhormat Kepala B2 P2 TOOT Bapak Ahmad Saiku MSc, yang terhormat Ketua LPPM Dr. Lusmeila Afriani, yang terhormat kepada seluruh dekan Universitas Lampung, Yang terhormat kepada seluruh pembicara pleno, yang terhormat Ketua Pelaksana Acara Ibu Endang Lini Widiastuti PhD, yang terhormat kepada seluruh peserta dan pemateri, dan kepada para hadirin sekalian yang telah hadir di acara pagi hari ini.
Welcome to the opening of International Seminar Traditional Herbal Medicines, Indonesian Medicinal Plants under the theme Future Research and Technology on Herbal Medicine Application for Diabetes and Other Degenerative Disorder.
Selamat datang di acara pembukaan International Seminar Traditional Herbal Medicine Indonesia Medicinal Plant dengan tema Future Research and Technology on Herbal Medicine Application for Diabetes and Other Degenerative Disorder.
Good morning, ladies and gentlemen. My name is Muhammad Aryan Hidayat and accompanied by my partner, Diandra. It is outstanding chance for us to be your master of ceremony in this morning on Thursday, 8th of July, 2021. Before we begin our seminar today, let me deliver the list of agenda.
Selamat pagi hadirin sekalian, nama saya Diandra Ayu dan ditemani oleh rekan saya Muhammad Rian Hidayat. Ini merupakan kesempatan yang luar biasa bagi kami untuk menjadi pembawa acara pagi ini pada hari Kamis, tanggal 8 Juli 2021. Sebelum kita memulai acara seminar pada hari ini, kami akan sampaikan susunan acara terlebih dahulu.
The first, opening ceremony with the special performance traditional dance. Second, singing the national anthem, Indonesia Raya. Third, report of the chairwoman of the event. Fourth, welcome speeches. Fifth, prayer. And the last, closing and photo session.
Pertama, pembukaan acara dengan pertunjukan tari tradisional. Kedua, menyanyikan lagu nasional Indonesia Raya. Ketiga, laporan Ketua Seminar TOI ke-59. Keempat, acara sambutan. Kelima, pembacaan doa. Yang terakhir, penutupan dan sesi foto.
The first agenda is traditional dance performance. So please welcome our special performance, Tari Sige Pengutan.
Acara pertama yaitu pertunjukan tari tradisional. Mari kita sambut pertunjukan spesial dari Tari Sige Pengutan.
Sige Pengutan dance is one of the new dance creation from Lampung area, Indonesia. This dance is a cultural fusion between two Lampung tribes, namely Pepadun and Saibatin. Besides being performed at a local traditional event through a religion regulation, the Sige Pengutan dance was inaugurated as the Lampung dance and welcomed important guests. Sige Pengutan dance has the meaning of contain the rain. The meaning of the movement contained the philosophy of Biil Pesenggiri. The property used in the Sige Pengutan dance, namely tepak, and its own meaning is used, the battle box will given to the one of the guests who is considered to the representative of the guest. This is the welcome and thank you from the host to the guests who have attended the event. Thank you very much for the awesome performance.
Terima kasih banyak atas pertunjukannya yang sangat luar biasa.
The next agenda is singing the national anthem Indonesia Raya. Ladies and gentlemen, please stand up.
Acara selanjutnya yaitu menyanyikan lagu nasional Indonesia Raya. Para hadirin diharapkan untuk berdiri.
Ladies and gentlemen, may take a seat.
Para hadirin dipersilahkan duduk.
The third agenda is speeches. First, we will hear the report by Chairman of the event, Ibu Endang Linirin Widyastuti, PhD. For Ibu Endang Linirin, time is yours.
Acara ketiga ialah laporan dari Ketua Seminar TOI ke-59. Untuk Ibu Endang Linirin Widyastuti, PhD, waktu dan tempat kami persilahkan.
Thank you. Tabi pun. Assalamualaikum warahmatullahi wabarakatuh. Good morning. Greeting for all of. May God bless all of us. First, I would like to welcome to all of you, to Honorable Rector of University of Lampung, main and keynote speakers, as well as moderators, presenters, and all guests and participants. At this moment, as a representative of the ISTHM 2021, I would like to give some report related to this event, the International Seminar on Traditional Herbal Medicine. We have already known that the theme of the seminar is the future research and technology on a herbal medicine application for diabetes and other degenerative disorder. The seminar is attended by 13 main and keynote speakers, which each individually is very expert and reputable in their field. This seminar also is attended by more than 350 participants, include 106 presenters with 95 oral presenters and 11 posters. All of them will also present their research related to the seminar theme. They are coming from many different prestigious universities from Indonesia, Malaysia, and from United States. For this, I express my thankfulness. This seminar is part of the National Seminar in Series by Indonesian Medicinal Plan, which is the 1959th seminar established by collaborative between the Research Community Services Institute of UNILA and the National Working Group of Medicinal Plant and Traditional Medicine, or we call it Pokjana Story. Last, we apologize for short notice and inconvenience that might happen during the seminar. We also ask permission for the head of the Center Research and Development of Medicine Plant and Traditional Medicine, which is B2P2TOT, or we call it B2P2TOT, to give some remark, and to the Director of UNILA, which is represented by the Vice Dean of Faculty of Mathematics and Sciences, to open this seminar. Thank you for your attention and cooperation. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much for Bu Endang Widya Suti.
Terima kasih banyak untuk Ibu Endang atas laporannya.
Next, we will hear the welcome speech by the head of Center for Research and Development of Medicinal Plants and Traditional Medicines. For Bapak Ahmad Saidku MC, times is yours.
Selanjutnya adalah sambutan dari Kepala B2P2TOOT untuk Bapak Ahmad Saiku. Waktu dan tempat kami persilahkan.
Thank you. Is my voice clear?
Yes, sir.
Okay, thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, selamat pagi, salam sejahtera, Om Swastiastu, Namo Buddhaya, salam kebajikan. Your Excellency, Rector of Lampung University, dear the main speaker, Director of Traditional Medicine Service, Ministry of Health, all keynote and invited speakers, Professor Taifu Mahmud, PhD, Professor Dr. Kamaluddin, Professor Dr. Asep Sukohar, MKes, Profesor Suharjo, PhD, Profesor Dr. Yuli Widyasuti, MP, Profesor Yana Maulana Shah, MS, PhD, Profesor Takuya Sugahara, Profesor Yaya Rukayadi, PhD, Profesor Dr. Apoteker Keri Lestari, MSc, Profesor Dr. Nur Hadian Ismail, and Dr. Masdaria Yunu Filsa Putra. Deans and Professor of Lampung University, all members of Working Group on Medicinal Plant and Traditional Medicine, also researchers, academic community, and all audience of the 59th Indonesian Medicinal Plant Seminar. Good morning and greeting from Tawangmangu. Alhamdulillah, praise and gratitude to Allah who has given time and all the goodness to all of us who are present online to take part in the 59th Indonesian Medicinal Plant Seminar in order to integrate the result of research on medicinal plant and traditional medicine in this country. This seminar is organized by the Institute for Research and Community Service of University of Lampung in collaboration with Working Group on Medicinal Plant and Traditional Medicine, or Pokjanas Atoi. Present the theme Future Research and Technology on Herbal Medicine Application for Diabetes and Other Degenerative Disorder. The committee designed this seminar to be an international event by involving a number of overseas speakers. Director of UNILA and all distinguished guests. The Working Group on Medicinal Plant and Traditional Medicine was born from a symposium held in 1985, which was later officially established with the name of National Working Group on Indonesian Medicinal Plant, or Pokjanas Toi, 31 years ago. Pokjanastoi, or no called Pokja TOT, was formed with the aim to coordinate research and development of medicinal plant and traditional medicine, covering upstream and downstream research activities, including research on medicinal plant conservation, cultivation, quality control, phytochemical to safety and efficacy. In addition, one of the working group activities is to schedule the National Seminar on Indonesian Medicinal Plants with two chosen medicinal plant as the main presentation. At the 59 Indonesian Medicinal Plant Seminar, the final Funiculum vulgaria and Yakon, Samalanthus sonsifolius Rob are the main object of the presentation. The theme of the seminar about research and technology of herbal medicine for diabetes is closely to Yakon, a medicinal plant that exhibits anti-diabetic activity, one of the plants chosen as the main theme. Fennel is known as a medicinal plant that widely used as carminative, digestive, lactagogue, and diuretic. This aromatic plant is used to treat the respiratory and gastrointestinal disorder as well. Its seeds are used as flavoring in packaged goods, meat and fish dishes, ice cream, and beverage. Phenol, phenolic glycoside, and aromatic volatile compound such as transanethol, estragol, and fenson are the major constituents of this species. Various in vitro and in vitro pharmacological experiments convincingly demonstrated the antifungal, antibacterial, antioxidant, antithrombotic, and hepatoprotector activities that support the phenol therapeutic function. The phenolic compound isolated from the phenol plant are thought to be responsible for its antioxidant activity, while the volatile compound make this plant an excellent flavoring agent. Currently, diabetes mellitus has become a serious health problem because it has affected more than 5% of the world's population. Some medicinal plants with anti-diabetic activity can be an alternative treatment for this chronic disease. Yakon is believed to have the potential to be developed as a choice of natural medicine for diabetes because it has hypoglycemic properties. Other pharmacological properties of Yakon are as antioxidant, prebiotic, anti-cancer, normolipemian, and cytoprotective. Yakon could be consumed by individuals with DM1 or DM2 or alteration of the syndrome metabolic due to its low caloric content. Moreover, it has preventive effect for the reduction of atherosclerotic and vascular risk factor in patients with hypoglycemic activity. The anti-diabetic activity of this plant has been preclinically tested. Yakon leaf ethanol extract was able to increase glycogen synthesis in the liver and muscle and could lower blood sugar. In another study, it was found that the extract of this plant improved the body weight and inhibited organ damage. Research on Yakon as a medicinal plant that has antidiabetic or hypoglycemic properties using leaves, root, or tuber have been carried out in many countries such as Italy, Brazil, Argentina, Japan, China, and Indonesia. The theme research and technology potential for diabetes chosen by the UNILA University Seminar Committee was the right choice. We know that diabetes is degenerative disease, which its prevalence continues to increase. The result of basic health research or CASDAS on 2018 showed that the prevalence of diabetes mellitus in Indonesia based on doctor's diagnosis at the age of 15 years is 2%. This figure show an increase compared to the prevalence of diabetes mellitus that was revealed by basic health research on the 2013 result of 1.5%. Based on data from the International Diabetes Federation or IDF, Indonesia in diabetes alert status because it ranked 7th of 10 countries with the highest number of diabetes patients. The prevalence of patients with diabetes in Indonesia reached 6.2%, which means that there are more than 10.8 million people suffering from diabetes per year on 2020. And endocrinology said that this figure is estimated to increase to 16 million patients per year on 2045. Ladies and gentlemen, in line with the theme Future Research and Technology on Herbal Medicine for Diabetes and Other Degenerative Disorder, which is carried out in this seminar, the Center of Research and Development of Medicinal Plant and Traditional Medicine, P2P2TOT, Ministry of Health, as the Secretariat of the Working Group and Medicinal Plant and Traditional Medicine is focusing on the synergy of accelerating phytopharmaca or phytomedicine. One of it is phytomedicine for diabetes. The synergy is active collaboration with traditional medicine industry, having ability to run a good manufacture traditional medicine for developing plant raw into phytomedicine. For this reason, we open the chance for equal position cooperation with four industry and research institutions that have a vision of developing phytomedicine. This phytomedicine development is in line with Presidential Instruction number 6 of 2016 concerning the acceleration of the and medical device industry. Some relevant government agencies are asked to take steps and collaborate to create an independent and highly competitive domestic pharmaceutical industry. This is confirmed by the issue of Ministry of Health Regulation number 17 of 2017 concerning the action plan for development of the pharmaceutical and medical device industry. The National Institute Health and Research and Development, Ministry of Health, has been transformed into the National Institute for Health Development Policy, PKPK. The mandate for P2P2TOT as one of the front line of PKPK is the recommendation on phytomedicine. All research activities should lead and support the production of phytomedicine or phytopharmaca. Therefore, it is necessary to accelerate the achievement of phytomedicine production, which will be recommended to primary healthcare policy holders. This phytomedicine product will be a great contribution from IHRD in responding to solution to health problem. In this implementation, phytomedicine product will also strengthen the existence of traditional health service, which can currently be operationalized under the legal basis of government regulation number 103 of 2014 concerning traditional health service and Ministry of Health Regulation number 21 of 216 articles, 5 paragraph, and concerning the use National Health Insurance Fund for health services and operational cost support in local government-owned primary healthcare. For this reason, on this occasion, as the chair of the working group on medicinal plant and traditional medicine, I invite academic and practitioners of traditional medicine, especially from industry, to jointly create a collaboration to obtain phytomedicine. I am optimistic that with the commitment of all parties in mutual research collaboration, by 2022, we will obtain 5 phytomedicine for at least 3 indications of degenerative disease, namely hypertension, diabetes, and hypercholesterolemia. Dear Rector of UNILA and all blessed seminar participants, on this nice occasion, I would like to express my deepest gratitude to the Rector of Universitas Lampung, the Head of Institute for Research and Community Service, LPPM, the Dean of Faculty Math and Science, and all the seminar committees who have prepared and realized this event very well. Thanks and highest appreciation to the speakers and all seminar participants for joining and contributing to this scientific activity. Finally, welcome to the 59 Indonesian Medicinal Plant Seminar. Hopefully, this will be our real contribution in the development of science and technology that support the promotive and preventive aspect of health through the use of medicinal plant and traditional medicine in Indonesia. Let me remind all attenders, let's take care of our health and obey the health protocol to prevent the spread of the COVID-19 disease. Hopefully, this pandemic will over soon. Bila taufiq hidayah. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Bapak Ahmad Saiku MSc, for the welcome speech.
Terima kasih banyak Bapak Ahmad Saiku untuk sambutannya.
Lastly, we will hear the welcome speech by the Rector of University of Lampung that will be represented by the Vice Dean of Academic and Cooperation, Faculty of Mathematics and Natural Sciences, Lampung University, Dr. Eng Heri Satria. And also, we like to have his honor to officially open this event. For Dr. Eng Heri Satria, time is yours.
Terakhir, sambutan dari Rektor Universitas Lampung yang akan diwakilkan oleh Wakil Dekan 1, Fakultas Matematika dan Ilmu Pengetahuan, Bidang Akademik dan Kerjasama, Bapak Dr. Eng Heri Satria. Dan juga kami ingin meminta kehormatan beliau untuk membuka acara ini secara resmi. Kepada Bapak Dr. Eng Heri Satria, waktu dan tempat kami persilahkan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
warahmatullahi wabarakatuh.
Ladies and gentlemen, in this blessed morning, please allow me to deliver our rector welcome speech. Head of P2P2TOOT, Dean of the Faculties in Universitas Lampung, Head of LPPM, Universitas Lampung, distinguished keynote and invited speaker, and all participants, the chairwoman of this seminar, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Tabi pun.
Iya pun.
Good morning. First of all, I would like to welcome all of you for participating in the international seminar on traditional herbal medicine, or ISTHM 2020, held in collaboration by Institute of Research and Community Service, or LPPM, Universitas Lampung, and Balai Besar Penelitian dan Pengembangan Tanaman Obat dan Obat Tradisional, or Center for Research and Development of Medicinal Plants and Traditional Medicine, the Ministry of Health Indonesia. The main theme of this year's seminar is Future Research and Technology on Herbal Medicine Application for Diabetes and other degenerative disorders. Thank you for participating in this international seminar. I would like to express my appreciation to our keynote and invited speakers, Dr. I Gede Made Wirabrata, SSI APT, MKes, MH, Director Yan Kestrat, Kemenkes, Professor Taifo Muhammad, PhD, Oregon State University, USA, Professor Takuya Sugahara, Ehime University, Japan, Profesor Dr. Yuli Widyastuti, BP2 TOOT, Profesor Dr. Nur Hadiani, UITM Malaysia, Profesor Dr. MT Kamaluddin MSc from Medicine Faculty of University of Sriwijaya, Profesor Dr. Kerry Lestari MSc IPT from Department of Pharmacy, University of Pajajaran, Professor Yana Maulana Shah, MS PhD from Faculty of Mathematics and Natural Sciences, ITB Bandung, Masteria Putra PhD from LON LIPI, Profesor Dr. Asep Sukohar MKes, Ketua ID Lampung from Faculty of Medicine, Universitas Lampung, and Professor Suharso PhD from Faculty of Mathematics and Natural Sciences, Universitas Lampung. We are honored that you would be participating in this event. Although we are now still in the COVID-19 pandemic situation, we are still able to participate in this seminar where all will be conducted through online system via Zoom meeting. In this century, diabetic and degenerative disorder have caused so many deaths to the human being in all parts of the world. The Health Society Security Agency of Indonesia has reported that based on the claim loaded by the user, diabetic and degenerative disorders such as cardiovascular disease are the most common reported disease in Indonesia. We have to be aware of the most of degenerative disorder almost have no cure, but medication and treatment can help improve countering symptoms, relieving pain, and improving mobility. Indonesia in general and Lampung in special case have so many traditional herbal medicine resources which can be future explored to find some new potent drug to help in the curing process of this disease. However, perhaps due to the some limitation of the available instrument in the laboratory, at most institutions in the Indonesia, the exploration of the traditional herbal medicine for this purpose is still quite limited. Therefore, the collaboration with institutions having state-of-the-art instrument is a must in order to maximize the potential use of the traditional herbal medicine. Thus, it is important to bring together experts in this field in hope that we can have better knowledge and produce innovation using the plant-based source of the traditional herbal medicine in Indonesia. Ladies and gentlemen, hopefully in this seminar, there will be sharing of new idea, discussion, collaboration between participants. I strongly believe that through this international seminar, we are able to improve our potency to face all challenges and achieve more opportunities in the future. I hope this seminar is able to inspire and deliver benefits to all participants. I believe that together we will be able to contribute to science and research, especially in the treatment of diabetes and other degenerative disorder using a variety of traditional herbal medicine, especially obtained in Indonesia. Once again, welcome to this international seminar and have a wonderful discussion. Thank you and have a great discussion. Wassalamualaikum warahmatullahi wabarakatuh. Professor
Waalaikumsalam.
Waalaikumsalam.
Karomani, Rector of University of Lampung. Ladies and gentlemen, with pleasure, with saying Bismillahirrahmanirrahim, this seminar open officially. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much for Dr. Eng Heri Satria for the welcome speech.
Terima kasih banyak kepada Bapak Dr. Eng Heri Satria yang telah membuka acara ini secara resmi.
Before we end this opening ceremony, let us say a prayer that will be led by Professor Sutopo. For Professor Sutopo, time is yours.
Sebelum kita mengakhiri upacara pembukaan ini, marilah kita panjatkan doa yang akan dipimpin oleh Bapak Profesor Sutopo. Untuk Bapak Profesor Sutopo, waktu dan tempat kami persilahkan.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Tabi pun.
Ya
pun.
Good morning, ladies and gentlemen.
Morning.
Let us bow our head for a moment while raising our hand to pray to Allah for the success of the ISTHM seminar 2021. Allow me to read the dua in the Islamic way. For those with other religion, please adjust according to your religion. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Wassalatu wassalamu ala asrofil anbiya wal mursalin wa ala alihi washabihi jema'in. Allahumma salli ala Muhammad wa ala ali Muhammad. Allahumma ighfir lil muslimina wal muslimat wal mu'minina wal mu'minat al ahya'i minhum wal amwat innaka sami'un qaribun mujibud da'awat wa yaqdiyal hajat. Allahumma arinal haqqa haqqa warzuqna tiba'ah wa arinal batila batila warzuqna tinabah. Ya Allah, Ya Rahman, Ya Rahim, You are the most merciful, the most beneficent. Oh Allah, today we academic community at the University of Lampung through the Institute for Research and Community Services in collaboration with the Center for Research and Development of Medicinal Plant and Traditional Medicine are holding the international seminar. For that, O Allah, grant us a success. Help us to make this event easy and help us so that this event will provide as much possible as benefit to humankind. Ya Allah, Ya Salam, You are the great giver salvation. Oh Allah, protect us from the dangers of the spread of this COVID-19 pandemic, which is still widespread everywhere, which is still happening anywhere. Ya Allah, Ya Ghafur, You are the most forgiving. We ask forgiveness from You. If in the organization of this seminar, there are things that might violate Your provision, Allahumma inna nas'aluka ilman nafi'an wa rizqan thayyiban wa amalan mutaqabbala. Allahumma inna nas'aluka al-huda wa tuqa wal afa wal ghina Allahumma inna na'udzubika minal barash wal junun wal zudam wa min sa'il asqam wa qarana khassah. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzabannar wa qina adzabannar wa qina adzabannar. Subhana rabbika rabbil izzati amma yasifun wa salamun alal mursalin walhamdulillahi rabbil alamin. Billahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Amen. May all God blessings come to us. Thank you very much, Professor Sutopo.
Amin, amin ya robbal alamin. Semoga kita semua diberkati oleh Tuhan Yang Maha Esa. Terima kasih banyak Bapak Profesor Sutopo.
Ladies and gentlemen, now we come to the end of the opening event of International Seminar Traditional Herbal Medicines Indonesia Medicinal Plants under the theme Future Research and Technology on Herbal Medicines Application for Diabetes and Other Degenerative Disorder. For closing opening ceremony, let us say Hamdalah.
Hadirin sekalian, sekarang tiba saatnya pada akhir acara pembukaan dari acara International Seminar Traditional Herbal Medicine Indonesia Medicinal Plan dengan tema Future Research and Technology on Herbal Medicine Application for Diabetes and Other Degenerative Disorder. Untuk menutup acara pembukaan ini, mari kita ucapkan hamdalah bersama-sama.
Next agenda is photo session. For all of the participants, may turn on the camera.
Acara selanjutnya yaitu sesi foto. Kepada para hadirin dipersilahkan untuk menghidupkan kamera.
Page 1. Page 2. Page 3. Page 4. Page 5. Okay, thank you very much for all of the participants.
Terima kasih banyak kepada para hadirin sekalian.
Now we will move to the main event, the plenary session. For the first plenary, that will be guided by Professor Suwijoyo Pramono as the first moderator. But before that, let me read his brief curriculum vitae.
Sekarang kita beralih ke acara utama yaitu sesi pleno. Untuk sesi pleno yang pertama akan dipandu oleh Profesor Suwijoyo Pramono sebagai moderator. Sebelum itu, izinkan rekan saya untuk membacakan kurikulum vitae singkatnya.
Professor Dr. Suwijoyo Pramono APT is a professor at Department of Paramedical Biology, Faculty of Pharmacy, Gadjah Mada University. Area of the expertise is medical and health science, phytochemistry, and also paramedical biology. His education background for undergraduate from pharmacy major and faculty, Gadjah Mada University from 1971 until 1976. For Master in Plains Pursuits Medicinal, Faculty de Science Pharmaceutics, University Paul Sabatier, Toulouse, France, from 1979 until 1980. And the doctor is the same with a master in the plates youth medical faculty de science paramedics university paul sabatier toulouse france from 1918 until 1982. Next, there are several his professional activity. The first, the lecturer of faculty of pharmacy Gajah Mada University from 1977 until now.
You can read only, yeah. You can read, not necessary. Okay, it is not matched with the screen, so you can read only of your hand. Yes. I think it's enough on the introduction.
Okay, thank you, Professor. So, ladies and gentlemen, please welcome our first moderator, Profesor Suwijoyo Pramono.
Hadirin sekalian, mari kita sambut moderator pertama kita, Profesor Suwijoyo Pramono.
Thank you very much, and I have to apologize because my name is not Suwijoyo but Suwijio. Thank you very much. Okay, well, we have now the main speaker, firstly, Dr. Wirabroto, and then we have two speakers in this session. The first one is Professor Taifu Mahmoud from Oregon State University, and then Professor Kamaluddin from Sriwijaya University. We enter now to the first speaker. It is main speaker, so we call Dr. Wirabrata, SSEPT plus MMH, and I have to look at the screen. Dr. Apotheker IKM Wirabrata. This I have to felicitate you as the new Direktur Pelayanan Kesehatan Tradisional. Dr. Wirabrata is from as one of Fakulty Farmasi Universitas Pancasila, and professional academy, I mean apotheker in the same faculty, Universitas Pancasila, and then S2 from Fakultas Kesehatan Masyarakat Universitas Indonesia, and then also he took S2 jurusan ekonomi Sekolah Tinggi Ilmu Ekonomi KB, and then the doctor, I mean S3 jurusan Ilmu Ekonomi Universitas Persada Indonesia JAI, and also extraordinary S2 from jurusan Hukum Pidana Universitas Pendidikan Nasional. And then we can look at the works experience. He is a Direktur Sumber Daya Manusia dan Pendidikan until 2018, yeah. And then Direktur Penilaian Alat Kesehatan dan Perbekalan Kesehatan Rumah Tangga until 2021, 4 June, yeah. And then from 4 June recently, yeah, 2021 until now as Direktur Pelayanan Kesehatan Tradisional. So Now, Dr. Wirabrata will speak about the use of medicinal plants to control non-communicable diseases in Indonesia. So you can speak in English or Indonesia. Do you welcome Dr. Wirabrata? Time is yours.
Thank you, moderator, Profesor Suwijo. Izin men-screen.
Yes.
Ya. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Salam sejahtera untuk kita sekalian. Om Swastiastu, Namo Buddhaya, salam kebajikan. Tabiku, Bapak Ibu sekalian, terima kasih yang saya hormati Bapak undangan, yang terhormat Bapak Rektor, Ibu Universitas Lampung, Dekan, Wakil Dekan, OC, Ibu Endang, ini PhD, Pak Ahmad Saikun, tadi menyampaikan sambutan, Ketua dan Pengurus Pokjanas, TOI, para narasumber yang terhormat, para guru besar, para klinisi, farmasis yang hadir, para tenaga kesehatan tradisional, pemerhati kesehatan, Bapak Ibu undangan, dan peserta International Seminar on Traditional Herbal Medicine 2021. Semoga kita dalam keadaan sehat dan ke depan tetap sehat, keluarga sehat. I'm sorry, my presentation in bahasa. Setelah presentasi saya juga izin, mohon maaf sekali lagi, karena masih standby situasi COVID zona merah di Jakarta. Berikut saya sampaikan terkait dengan penggunaan tanaman obat untuk mengendalikan penyakit tidak menular PTM di Indonesia. The use of medicinal plants to control non-communicable disease in Indonesia. Ya, tadi disampaikan Bapak Moderator, terima kasih, Prof. Bapak Ibu sekalian, yang saya hormati, para peserta pembangunan nasional dan pembangunan kesehatan nasional. Ini merupakan dasar dari beranjaknya Peraturan Presiden 18 2020 yang dinyatakan sebagai salah satu kebijakan dan strategi untuk meningkatkan pelayanan kesehatan menuju cakupan kesehatan semesta, terutama penguatan pelayanan kesehatan dasar primary health care dengan mendorong peningkatan upaya promotif preventif, mendukung inovasi dan memanfaatkan teknologi. Kalau dilihat di slide, peningkatan kesehatan ibu, anak, keluarga berencana, kesehatan reproduksi, percepatan perbaikan gizi masyarakat, kalau sekarang dikenal dengan bagaimana menurunkan stunting, kemudian peningkatan pengendalian penyakit, pembudayaan gerakan masyarakat hidup sehat, GERMAS, penguatan sistem kesehatan dan pengawasan obat dan makanan. Ini menjadi satu kesatuan pelayanan health delivery dengan penguatan preventif dan promotif. Tentunya penguatan by system, nah ini menjadi holistik, satu kesatuan di negara kita menggunakan ini. Tentunya ini menjadi satu kondisi ketika pandemi terjadi, ini sangat-sangat berpengaruh pada sistem yang ada, dan kita perlu penguatan-penguatan dalam strategi ketahanan nasional. Bapak Ibu sekalian, permasalahan kesehatan saat ini, memang kalau ini perspektif saja dari beberapa peneliti, bahwa 70% yang merasa sehat tadi akan berada di, katakanlah di sebelah kanan, kemudian 30% yang merasa sakit, selanjutnya dari 30% tadi akan berusaha untuk mencarikan pelayanan kuratif sebesar 52%. Ini masuk ke puskesmas, lini pertama di pelayanan JKM. Nah, dari sini termasuk yang nanti masuk ke rumah sakit setelah mendapatkan informasi bahwa ini harus dirujuk untuk mendapatkan pelayanan rujukan. 48% itu mengobati sendiri, self-care. Ini sebenarnya targetnya. Di kesehatan tradisional ini menjadi target. Kalau dilihat dari 70% rasa sehat, ini kembali ke paradigma sehat tadi. Mainkan di situasi bahwa ada geria sehat namanya. Suatu lembaga berdiri sendiri di luar dari rumah sakit ataupun puskesmas yang menyelenggarakan kesehatan tradisional. Nah, kesehatan tradisional inilah yang sebenarnya Bapak Ibu sekalian turun-temurun ini. Nah, ini yang turun-temurun yang saya kira menjadi satu yang kebanggaan kita juga, melanjutkan apa yang sudah ada sejak dahulu kala untuk terus bisa ditingkatkan dalam rangka meningkatkan kesehatan masyarakat yang setinggi-tingginya. Tentunya usaha ini terkait dengan upaya promotif preventif dan pemulihan pasca pengobatan. Kalau dilihat dari riset kesehatan dasar 2013 dan riset kesehatan dasar untuk 2018, ada peningkatan, Bapak Ibu. Ada peningkatan persentase masyarakat menggunakan pelayanan kesehatan tradisional. Ini bagus sekali, karena udah 44,30% masyarakat menggunakan itu. Berarti proporsi pemanfaatan upaya tradisional ini meningkat dengan melakukan survei besar ini. Nah, kalau dilihat jenis tenaga kesehatan kita yang nakes tradisional dan hatra ini masih terbatas memang jumlahnya. Kampus-kampus yang memiliki fakultas kesehatan tradisional juga masih terbatas. Ke depan ini adalah peluang. Bagaimana nanti kampus-kampus akan melahirkan tenaga kesehatan tradisional baru. Kami juga di pemerintah, di Kementerian Kesehatan, sedang mengupayakan jabatan fungsional untuk tenaga kesehatan tradisional. Nanti akan muncul kelompok atau jenis tenaga kesehatan baru, yaitu tenaga kesehatan tradisional. Nah, ini memang diharapkan kalau ini bisa lahir, tentunya bisa membantu kesehatan nasional secara menyeluruh dan utuh. Yang melatar belakangi pemanfaatan obat tradisional. Indonesia kaya akan bahan baku. Jelas, banyak sekali. Ya, kalau cerita nanam apapun di Indonesia tuh hidup, kan gitu. Nah, ini menarik. Permintaan kebutuhan obat tradisional terus meningkat. Situasi pandemi, percaya tidak percaya, mungkin Bapak Ibu sekalian, di sini banyak juga yang minum jamu. Saya minum jamu tiap hari. Terkait dengan meningkatkan imunitas tubuh. Saya kira Bapak Ibu sekalian yang mempelajari itu, ataupun masyarakat luas, juga percaya tentang itu, akan memanfaatkan ini. Selanjutnya adalah pengembangan ISTHM dalam produk obat tradisional, mendorong pertumbuhan ekonomi dan produk industri obat tradisional. Selanjutnya yang penting nanti tahun depan adalah OMAI, obat modern asli Indonesia, di mana mempunyai khasiat yang hampir sama dengan obat kimia sintetik. Tentunya nanti melalui tahapan-tahapan uji klinik yang dapat dipertanggungjawabkan. Gitu. Riset tanaman obat dan jamu, saya kira Kristosja dari tahun ke tahun, ada 2012, 2015, 2017, temuan-temuannya banyak sekali yang bisa dimanfaatkan. Bapak Ibu sekalian, tentunya spesies tanaman obat banyak di laut, di darat, jumlah populasi BPJS ini menjadi peluang besar kita. Bahwa kesehatan tradisional, obat tradisional, jamu, itu akan sangat berperan nantinya untuk membantu kesehatan dari sisi promotif preventif. Bahkan, pasca pelayanan kuratif. Jadi, pada saat pengembalian pemulihan kesehatan dari pasien. Ini besar peranannya. Kalau dilihat dari sisi WHO Traditional Medicine Strategy sampai 2023, untuk produk, practices, dan practitioner rate, di jamu itu udah ada 11.000, Bapak Ibu. Ada 11.000 produk. OHT ada 17 produk. Fitofarmaka 25 produk. Ini adalah kebanggaan, menghasilkan hasil-hasil temuan dari para peneliti yang hadir di sini, ini membanggakan. Kedepannya bagaimana? Kedepannya tambahkan lagi temuan-temuan itu. Selanjutnya kami nanti akan mencoba apakah fitofarmaka yang telah melalui uji klinis tadi bisa masuk nanti ke pelayanan JKN misalnya. Sekarang sudah masuk beberapa. Nanti kalau bisa kembangkan lagi, ini menjadi satu peluang besar, Bapak Ibu sekalian. Jangan dikotomikan, memisahkan antara konvensional medicine dengan traditional medicine. Tapi bagaimana caranya ini berisi. Bisa jadi pengganti, bisa komplementer, bisa saling melengkapi, dan sebagainya. Gitu. Karena ini sudah ada. Di Indonesia ini sudah ada. Ini yang perlu kita kembangkan bersama. Tentunya para guru besar, para pakar yang hadir di sini, sepakat bahwa ini adalah asli Indonesia yang patut kita bisa kembangkan selanjutnya. Selanjutnya, Bapak Ibu sekalian, saya ingin sampaikan bagaimana peluang ke depan bahwa obat OMAI ini nantinya digunakan untuk menggantikan kekosongan ketersediaan obat kimia. Nah, jadi kalau tidak ada obat kimianya, masuk obat tradisional. Selain adjuvat misalnya pada kanker, digunakan untuk efek samping kemo. Ini bagus untuk meningkatkan quality of life. Gitu. Melengkapi pengobatan konvensional sebagai pendamping on top obat kimia, misalnya obat diabetes, lupus. Pemanfaatan obat tradisional sejak zaman dahulu merupakan tradisi. Nah, ini turun-temurun, Bapak Ibu sekalian. Local wisdom. Saya yakin di mana-mana tempat Bapak Ibu mengikuti Zoom ini pasti ada ciri khas obat tanaman keluarga yang bisa dikembangkan di sana. Di Lampung, apa hasil budidaya yang bisa dikembangkan ke arah sana. Ini nantinya akan meningkatkan akses dan mutu pelayanan kesehatan tentunya. Kita sebagai pemerhati kesehatan di sini tentunya berharap hasil-hasil budidaya yang ada di daerah masing-masing dapat dimanfaatkan untuk sebesar-besar untuk kepentingan masyarakat. Tentunya paling tidak masyarakat setempat itu bisa mengoptimalkan pelayanan yang ada. Bapak Ibu sekalian, kalau lihat regulasi, saya kira regulasi sudah banyak. Nah, ini penting, seperti Peraturan Menteri Kesehatan nomor 21 2016 tentang penggunaan dana kapitasi JKN untuk jasa pelayanan kesehatan dan dukungan biaya operasional pada SKTP milik pemerintah. Di situ dijelaskan bahwa dalam hal obat dan bahan medis yang dibutuhkan dan tidak tertentu dalam formulir nasional sebagai masuk pasal ayat 5, dapat menggunakan obat lain, termasuk obat tradisional, jelas. Obat herbal terstandar, fitofarmaka secara terbatas, dengan persetujuan kepala dinas. Artinya di regulasi sudah ada, gitu. Sudah bisa nyantol, payung hukumnya sudah jelas. Di Permenkes nomor 85 2019 tentang petunjuk operasional penggunaan dana alokasi khusus fisik bidang kesehatan tahun anggaran 2020, pemilihan jenis obat vaksin mengacu pada daftar esensial di Fornas, sedangkan BMAP mengacu pada daftar alat kesehatan non-elektromedik pada kompendium alat kesehatan. Serta pedoman teknis yang ditetapkan melalui peraturan pemerintah atau peraturan menteri dalam hal obat BMAP tidak tercantum acuan di atas tadi, dapat digunakan obat dan BMAP lain sesuai dengan termasuk obat tradisional, fitofarmaka, dan obat herbal terstandar secara terbatas. Nah, ini sebenarnya peluang ke depan bagaimana menghasilkan formularium herbal misalnya, formularium obat tradisional. Buat listnya. Kita akan sama-sama. Begitu. Ini peluang ke depan Bapak-Ibu sekalian, para peneliti yang di bidangnya perdalam. Kami perlukan uji kliniknya. Informasikan ke pemerintah untuk bisa ditindaklanjuti dalam bentuk regulasi. Itu penting. Setelah diregulasikan, nanti tenaga kesehatan lainnya, para klinisi, farmasis, tenaga kesehatan tradisional dapat memanfaatkan itu di fasilitas pelayanan kesehatan. Itu alurnya, sehingga bisa implementatif apa yang oleh para peneliti di sini. Termasuk juga peraturan menteri lain yang saya kira terkait yang dapat di pemanfaatan obat tradisional. Ini ada jamu terregister. Moral Asli Indonesia nih. Karena udah banyak, uji kliniknya udah ada. Ya gitu ya. Nah, ini bisa dimanfaatkan. Nanti akan menjadi formulirnya kabupaten kota. Kami tingkatkan lagi nanti menjadi formulirnya di tingkat pusat. Supaya dari Sabang sampai Merauke. Begitu. Sehingga nanti dokter menerjemahkan promotif, preventif, kuratif kemana wilayahnya. Nah, ini optimalkan dalam regulasi. Selanjutnya, Bapak-Ibu sekalian yang saya hormati, seminar Internasional 21 2008 tentang standar. Kalau dilihat, pelayanan medik herbal bisa dilakukan oleh dokter, dokter gigi, dokter spesialis. Tentunya dasar yang mempunyai sertifikat kompetensi penting karena memang menjadi kemampuan khusus. Jadi ada dokter yang melakukan pelayanan medik kode etik profesi, tentu pasti dan tidak bertentangan dengan kode etik kedokteran Indonesia. Begitu. Dalam pelaksanaan di fasilitas pelayanan, ada obat jadi, ada meracik sendiri. Kalau meracik sendiri, dokter melaksanakan pelayanan perlu didampingi asisten apoteker sebagai farmasis yang membantu peran sediaan obatnya. Nah, ini ada diatur di Permenkes 3 2010 tentang sertifikasi jamu sebagai bentuk penelitian berbasis pelayanan. Ini sudah jalan. Kalau ini sinergi para klinisi, farmasis, nakesstrat, jalan. Ini akan sangat-sangat bermakna bagi masyarakat. Oh my, ada geria sehat, ada rumah sakit, dan puskesmas. Kami mencoba menjelaskan secara terpisah bagaimana geria sehat itu bisa lahir, berdiri sendiri dia. Ada tenaga kesehatan tradisional sebagai pelaksana SDM di sana. Ada nakesstrat secara profesional di sana. Ada standarnya di sana. Kemudian melakukan persiapan OMAI. Sedangkan di Fasiankes, di rumah sakit dan puskesmas, ada dokter di sana, ada DPJP-nya, dokter menjawab pelayanan, di sana ada, di rumah sakit. Nah, di peningkatan kompetensi dokter dalam pemanfaatan omai tadi, kemudian resepkan. Sama-sama, tujuannya bisa dioptimalkan pemanfaatan omai bagi pasien dan masyarakat. Prinsip pemanfaatan obat tradisional. Pertama adalah mengacu pada kepentingan terbaik pasien. Tentunya ditanya, informed consent harus jalan. Nah, ini secara hukum harus. Kemudian yang kedua adalah pimpinan Fasiankes, pimpinan rumah sakit atau puskesmas berwenang menetapkan jenis obat tradisional digunakan. Nah ini boleh berarti dirut rumah sakit boleh menetapkan itu. Diutamakan sebagai promotif preventif. Harus aman, berkhasiat dan bermutu. Itu pasti wajib itu. Nah, termasuk penggunaan obat tradisional yang terregistrasi oleh Badan POM sebagai instansi lembaga negara yang punya kewenangan untuk memberikan izin. Obat tradisional yang bersumber dari hewan harus memiliki sertifikat halal. Harus, ini harus. Tidak dalam bentuk simplisia, kecuali dalam rangka penelitian berbasis pelayanan. Emergency tidak boleh digunakan dalam keadaan gawat darurat dan keadaan yang potensial membahayakan jiwa. Saya kira Bapak Ibu sekalian yang hadir di sini memahami bagaimana prinsip pemanfaatan obat ini, supaya bisa bagi tenaga kesehatan mudah melaksanakannya, bagi pasien juga mendapatkan keamanan, khasiat, dan efikasi yang bermutu dari produk, dan pelayanannya tentunya. Bapak Ibu sekalian, target pencapaian Renstra kami di pemerintah adalah jumlah puskesmas dan jumlah sakit yang melaksanakan integrasi pelayanan. Jadi pelayanan konvensional dengan pelayanan tradisional itu terintegrasi di pelayanan. Itu tugas kami juga. Mudah-mudahan ke depan target demi tahun bisa tercapai, banyak rumah sakit yang akan menyelenggarakan ini, dan tentunya akan menjadi pilihan alternatif juga para klinisi untuk memberikan pengobatan atau alternatif tindak lanjut dalam pengobatan pasien. Selanjutnya kami juga punya geria sehat, Bapak Ibu sekalian. Mungkin di Lampung nanti ada pengembangan geria sehat, atau Bapak Ibu di mana Prof. Soswi mungkin di Jogja banyak obat tradisional yang kita buat geria sehat. Nanti ada akupunturnya, ada akupresurnya, ada pelayanan jamu. Nah, ini menarik sekali. Di kantor Kementerian punya. Nah, terima kasih, Prof. Ini menarik karena sayang kalau di jalan Pemdanya yang menyiapkan fasilitasnya, kami siapkan standar regulasinya, tentunya nanti gubernur dan bupati kota, wali kota akan menyiapkan regulasi. Selanjutnya adalah strategi intervensi pada penyakit tidak menular. Ada perlindungan khusus, ada penanganan kasus. Di sini yang akan lihat bagaimana multisektor akan terkolaborasi. Setelah kami di alat kesehatan waktu pada awal pandemi, kita hilang masker waktu itu ya, sulit sekali mencari masker. Saya sempat ditanya ke pimpinan bagaimana ini masker saya hilang. Banyak sekali barang impor yang masuk, termasuk adalah bahan baku masker. Nah kalau dilihat di sini, Bapak Ibu sekalian, perubahan perilaku masyarakat, meningkat untuk menjadi pelayanan kesehatan tradisional, untuk mendeteksi dini penyakit tidak menular, ada diabetes, ada intervensi, nah ini kita bisa jaga itu. Nah, kalau dalam perlindungan tentunya kita vaksinasi, termasuk yang COVID sekarang ini kita lakukan vaksinasi. Dan selanjutnya adalah penanganan kasus dengan pengobatan di fasilitas pelayanan kesehatan sesuai dengan standarnya. Dan sekalian yang saya hormati, prioritas saat ini ada populasi sehat, ya, ada kondisi populasi berisiko, ada penyakit tidak menular dan penyandang penyakit tidak terkontrol. Ini coba di kluster, sebenarnya tidak memisahkan, tapi mencoba mengkluster pemisahan bagaimana primary prevention yang akan dilakukan di populasi sehat. Relasi ini kita pertahankan tetap sehat, kan gitu ya. Upaya kesehatannya seperti apa pemutihan. Selanjutnya pada populasi berisiko, secondary prevention dan early detection, kirim dulu. Mudah sekali, untuk kalian ini sentimeter, uji efek, ya, ada institut diagnostik untuk gula, kolesterol, hasilnya seperti apa. Secara berkala, tentunya dengan menambahnya usia, kita perlu melakukan tes itu. Kemudian akan dilakukan intervensi dini dan kontrol. Mudah-mudahan belum jadi tahu berat. Selanjutnya adalah apabila kekayaan ini menjadi penyakit yang perlu diobati, termasuk nanti perawatan berkelanjutan. Ini sampai di tingkat yang lebih advance, bagaimana pelayanan itu lebih komprehensif lagi. Sayang kan kalau dari warna merah ini. Diharapkan warna kuning ada, tuh kita bisa reduksi dan menjaga kondisinya tetap terkontrol. Selanjutnya, teman-teman sekalian, saya ingin menyampaikan tentang pemanfaatan omega tadi untuk hipertensi, komplikasi kronis dan waktu dan kolesterol tinggi. Sebenarnya di WHO sendiri tahun 2015 menunjukkan bahwa 1,13 miliar orang di dunia menyandang hipertensi. Konon kabarnya 1 dari 3 orang di dunia terdiagnosis hipertensi. Jadi 1 dari 3. Ini luar biasa. Jumlah penyandang hipertensi terus meningkat setiap tahunnya, diperkirakan tahun 2025 1,5 miliar orang menyandang hipertensi. Dan diperkirakan 9,4 juta orang meninggal akibat hipertensi dan komplikasinya. Institute for Health Metrics and Evaluation tahun 2017 menyebutkan bahwa dari total 1,7 juta kematian di Indonesia, didapatkan faktor risiko menyebabkan kematian adalah tekanan darah atau hipertensi sebesar 23,7%. Hipertensi sekarang jadi masalah utama kita, tidak hanya di Indonesia dan dunia, karena hipertensi merupakan salah satu pintu masuk faktor risiko penyakit jantung, gagal ginjal, diabetes, dan stroke. Riset 2015 menyatakan prevalensi hipertensi di silent killer berdasarkan hasil pengukuran pada penduduk usia lebih besar dari 18 tahun sebesar 24,1%. Kalimantan Selatan 44,1%, sedangkan terendah di Papua sebesar 22,5%. Berdasarkan data riset ini, jumlah orang Indonesia usia 15 tahun ke atas memiliki diabetes hingga tahun 2019 mencapai 11 juta jiwa. Besar sekali kan. Nah, ini yang perlu kita waspadai, sehingga memiliki kadar kolesterol tinggi, batas normal untuk clinical research support Fakultas Kedokteran Universitas Indonesia sekitar 35% penduduk Indonesia memiliki kadar kolesterol lebih tinggi. Ini yang saya kira data-data yang dari riset kesehatan dasar bisa dianalisis lebih jauh, bagaimana dilakukan kajian-kajiannya, analisis mendalam, itu jadi dasar hybrid atau dasar dengan kedua. Ini adalah memanfaatkan toga dan akupresur di populasi. Nah, ini memelihara kesehatan bugar, mengatasi gangguan ringan. Nah, bagaimana yang bugar tadi tetap bugar, yang ringan tadi menjadi bugar. Nah, tentunya mengurangi faktor risiko, meningkatkan daya tahan tubuh yang lebih baik. Ini tanaman obat keluarga. Saya kira di kecamatan kelurahan itu masih berjalan itu tanaman keluarga itu. PKK-nya saya kemarin beberapa zooming melalui di Kalimantan, di Sulawesi juga masih berjalan dengan baik. Istri Bupati Wali Kota yang biasanya ketua-ketua PKK ini sangat antusias untuk bisa pertahankan di Indonesia dalam kerja ini dan juga peningkatan kultur. Ya, kita pegang-pegang sedikit, kadang-kadang pijit-pijit hilang, pusing pijit-pijit itu satu upaya. Saya kira Bapak. Yang ini saya kira bisa dilestarikan. Selanjutnya ini adalah Bernard Bude Jamu, saya kira ini sebagai ikon saja bahwa jamu Indonesia, ayo kita minum sama-sama, ada budenya Bude Jamu, dan itu adalah ada gerainya. Di Kementerian Kesehatan ini ada, saya kira di Rawamangu juga sertifikasi jamu dan seterusnya, Bapak Ibu di tempat kerja masing-masing juga ada mungkin jamu-jamu yang disiapkan. Saya kira ini bagus. Selanjutnya, sekalian ini mengakhiri menyiapkan sistem pelayanan kesehatan agar obat tradisional dapat diterima di fasilitas pelayanan kesehatan. Pertama adalah mengembangkan dan meningkatkan obat tradisional itu untuk mendukung yang kedua adalah sosialisasi advokasi Permenkes 37 tentang pelayanan kesehatan tradisional terintegrasi antara konvensional dengan tradisional, dia akan menjadi terintegrasi. Nah, itu yang ketiga adalah revisi 121 tentang obat herbal di pasien agar pemanfaatan omai tadi dapat diimplementasikan. Keempat adalah membuat seminar bekerjasama dengan ID, industri farmasi dan perguruan tinggi lainnya, termasuk farmasi mungkin di sini bisa dikembangkan untuk memanfaatkan obat omai. Kolaborasi antara peneliti dan industri ini penting, karena selama ini antara obat tradisional, herbal, alat kesehatan bahkan itu kolaborasi antara peneliti dengan industri ini penting. Karena industri yang akan melakukan komersialisasi dan hilirisasi sampai terjadinya produk sampai isi, sedangkan peneliti menghasilkan satu temuan-temuan baru yang mungkin perlu dijembatani. Nah, ini yang penting. Nomor 5 ini penting standarisasi bagi para peneliti. Jangan berhenti, setelah ketemu, informasikan, dijurnalkan boleh, dan seterusnya. Lanjutkan ke tahap berikutnya adalah ketemu siapa industri yang cocok. Nah, cocok industrinya untuk melakukan komersialisasi tingkat besar. Jadi, produksi besar, kualitas tetap sama, bisa dimanfaatkan. Keenam adalah mewujudkan suatu kondisi harmonisasi investasi dalam rangka sinergi. Ini akademisi, bisnis, government, saya kira pertemuan seperti ini juga kami dari pemerintah sangat mendukung. Terima kasih undangannya dan kolaborasi kita. Tentunya nanti melibatkan juga pihak swasta dalam mengembangkan tema ini. Demikian saya kira yang bisa saya sampaikan. Terima kasih. Mohon maaf kalau ada hal yang kurang berkenan. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Kembali ke moderator. Terima kasih, Prof.
Waalaikumsalam warahmatullahi wabarakatuh. Alhamdulillah, luar biasa sekali Pak Dr. Wirabrata, sangat-sangat jelas. Yang saya catat tadi yang sangat menunjukkan pada waktu membaca beberapa SK yang keluar kemarin, para dokter agak sedikit gelisah. Itu apa? Jangan-jangan nanti dokter tidak boleh menggunakan obat tradisional. Tapi ternyata sekarang kita muncul dari orang pertama yang punya itu. Ternyata nanti akan ada jalur-jalur yang jelas sekali. Jadi ini yang sangat menyejukkan, sehingga kami mengharap Dr. Wirabrata terus menjaga kesehatannya, karena saya lihat pekerjaan berikutnya sangat banyak. Insya Allah kita semuanya akan mendukung. Kita beri aplaus kepada Dr. Wirabrata. Terima kasih. Semoga semuanya menjadi lebih jelas dan lebih bekerja sama mendukung apa yang menjadi program pemerintah. Terima kasih. Saya persilahkan apabila nanti ada tugas-tugas lain yang segera akan dilakukan. Terima kasih. Ladies and gentlemen, I hope you enjoy with this lecture. So we can look at now the agenda. The second speaker and this invited speaker, the first invited speaker is Professor Taiful Mahmud, PhD. He is a professor of the Department of Pharmaceutical Sciences, Oregon State University, Oregon Health and Science University, USA. Please, we can look at the education and training. Oh, extraordinary. He was graduated from Faculty of Pharmacy, University of North Sumatra Medan. Oh, so extraordinary. And then he takes MSc and PhD of the Faculty of Pharmaceutical Sciences, Osaka University, and then University of Washington Seattle during 4 years, 3-4 years postdoc, in bioorganic chemistry. And I cannot look at the rest. So okay, and then so please share the screen. Organizing committee, please share the screen. Okay, but okay, extraordinary the position and employment. But I want to look at you can read after the seminar. But the honor and award, there are 8 honor and award. But if we look at the appointment of Professor Taiful, there is a managing director of International Indonesian School Association in the US. So this is a very important person for students who want to study abroad, especially in the United States. And then he has 35 from the Google Scholar Age Index, 3 book chapters, and more than 250 conference abstract and invited lectures. Extraordinary. And patent awards and invention, he has a pen. So today, because you know, diabetes, there is a preventive and curative. One say that preventive is preferable. I mean, it's more important than curative. Today, Professor Taifur Mahfud will speak about the alpha glucosidase from inhibitors, alpha glucosidase inhibitors from plants and bacteria. 25 minutes time is yours, Professor.
Thank you, moderator, Professor Suwijio, for the kind introductions. First of all, I would like to thank the organizer for inviting me and giving me the opportunity to talk here. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
So I look at the theme of this international conference, which is about diabetes and other degenerative disorders. So I thought I would share with you some of the work that we have been doing with alpha glucosidase inhibitors. As you know, alpha glucosidase inhibitors are being used as anti-diabetic drug as well. So please allow me to share my slide. All right, so diabetes is one of the most prominent disease that is on rise worldwide right nowadays because of the rising levels of obesity, physical inactivities, poor diets, a lot of gadget, I guess, and work from home perhaps would contribute to diabetes in the future. And it has been estimated that, well, more than 420 million people have diabetes in the world. And in 2017, over 10 million cases of diabetes in Indonesia. That number was reported cases. You know, who knows, the one that have not been reported or are not reported, perhaps twice as many as that. So diabetes obviously a really problem that we have in the world as well as in Indonesia. Luckily, there are a number of drugs that can be used to treat diabetes, right from the very early drug like sulfonylurea, and then later on meglitinides that stimulate insulin release. And then also we have biguanides, which basically the ideas come from natural products, which improve insulin sensitivity and reduce hepatic glucose production. And then later on, we also have thiazolidinediones group of compounds that improve insulin sensitivity. So some type 2 diabetic patients, it's not because they cannot produce insulins, but sometimes the insulin sensitivity is very low. But the problem with those drugs are, some of them have pretty significant side effects. Some of the thiazolidinediones drugs also were taken out of the market because of the cardiovascular side effect. Then later on, nowadays we have more advanced drugs, and they are very good. Some of them are peptides that can mimic GLP-1 enzymes. Some of them also proteins that can mimic amylins, and some inhibitor can inhibit enzyme that degrade GLP-1s. And there is another group of anti-diabetic drugs that we call alpha glucosidase inhibitors. So these drugs inhibit the alpha glucosidase enzyme in the intestine or stomach and prevent absorption of glucose or monosaccharide into the blood. So that cause, that basically lead to the lowering of blood glucose. So this drug seems to be, much less side effect, presumably because it does not really directly involve in the body, but rather just inhibit the enzyme that digests carbohydrates. And because we are talking about herbal drugs here, so actually there are a number of plants or herbal that have been used for centuries that contains alpha glucosidase inhibitors. So one very most prominent one from the plant Salacia reticulata or Salacia oblonga or Salacia sinensis. This plant or class of plants, they produce very interesting compound that we call pyosugar sulfonium sulfates. And this plant has been used widely in India, Sri Lanka, in Southeast Asia as well. In Ayurvedic drug or medicine, sometimes they make this cup from the wood of this plant and then put water in the cup and wait or let it stand for an overnight, and then the next morning they would drink the water. Because these compounds are water soluble, currently they can extract the compounds into the water that they fill in into the glass. So definitely the chemical ingredient of the plants really support the use of the plant as anti-diabetic drugs. And then another group of alpha glucosidase inhibitors we call immunosugars. And many plants actually contain these immunosugar type compounds. One, the most prominent one is Morus rubra or mulberry. Mulberry, obviously, many people know is being used to feed silkworm. And apparently silkworm can, enjoy these mulberries, but the plant itself is very toxic to other insects. So by some reason, silkworm is resistant to this plant. This plant produces latex that contains a lot of immunosugar molecules, like 1-deoxynogermycin or deoxymannonogermycin and many others. So this just a few of them, actually many of this type of molecule, which is very interesting because I wonder why plant produce so many sugar-like molecules. They are not really sugar because there's no oxygen here, but rather amino group. And the other group, the compound that I show you just now, actually contains sulfur. So we call them as sugar mimetic or pseudosugars. And interestingly, bacteria also produce a lot of immunosugar. So presumably the structure is this one is a privileged structure that many organisms like to produce them, although we don't really know why they produce those compounds. So like Bacillus and Streptomyces produce these molecules. And human is taking advantage of that by converting the oxynogermycin to make compound called miglitol, which is an anti-diabetic drugs. And then some derivatives called miglustat is being used to treat Gaucher disease type 1, which because it inhibits glucosylceramide synthase. Because this type of compounds are produced by bacteria, that really give us opportunities to study the biosynthesis of those compounds more in more details, because you can get the sequence of the genome of the bacteria very quickly. So you can look at the blueprints of the bacteria and find the genes that are involved in making the molecule. So apparently, even though I just give you 3 examples here, but many of them, even though the organization of the genes in the genome or in the chromosomes, they are different, but all of them consistently have 3 genes, conserved genes that are involved in making these molecules. So another group of alpha glucosidase inhibitors that are that is very important is pseudo-oligosaccharides. Oligosaccharides, many of you know the meaning of oligosaccharides, but in this case, we call it pseudo-oligosaccharide because it's not real oligosaccharide. It's contain sugar mimetic or compounds that just resemble sugar, looks like sugar, like this, but there is no oxygen in the ring. So in this case, some people call carbo sugars. One example is Alcabos, which is an anti-diabetic drugs produced by Bayer, and then validamycin A, which is also produced a lot and being used as antifungal agent to treat plant disease, and many other pseudo-oligosaccharides are produced by bacteria, which again, very puzzling why they even produce this kind of compounds. And Acabos, as I mentioned, is being used as anti-diabetic drugs, was discovered many years ago by Bayer company. It's produced by the soil bacterium Actinoplanus species, but other bacteria like Streptomyces also produce this type of compounds. And you can find them in on the market like Glucobi, or in the US and Canada, we call the product Precos. So the way how this molecule or this compound work is Acabos inhibit the enzyme amylase. As you know, amylase hydrolyze amylin or starch, carbohydrates, right? And there are a lot of pockets in the enzyme, and then there's hydrolytic point here that hydrolyze the glycosidic bonds. The way how it does it by using these basic residues that attack the carbonyl, the carbon here, the olefinic, the glycosidic carbon, and then break open this bond, and then form this intermediate oxocarbenium transition state. So you see it changing from chair form to the half chair form here. So apparently Acabos resemble or mimic this transition state and bind to the enzyme very strongly. And then the enzyme cannot hydrolyze it because it has the CN bond instead of CO bond. So the Ki value is very, very low compared to carbohydrates. So that's the way how Acabos competitively inhibit the enzyme and making the enzyme cannot digest carbohydrate anymore. And of course, because of that, it has been proposed, or maybe Acabos even can be an alternative to metformin for first-line treatment in type 2 diabetes, because by inhibiting alpha-glucosidase enzyme, that would reduce postprandial glucose excursion and would delay distal intestinal absorption of carbohydrate, and in the end will increase GLP-1. Now, if you increase GLP-1, that of course you can reduce glucagon and reduce hyperinsulinemia and delay gastric emptying. And then also GLP-1 can also work in the nervous system to increase satiety or reduce appetite, and then you will get loss of weight. So that's the reason why Acabos looks like pretty good anti-diabetic drugs. But the problem is sometimes Acabos also can alter gut microbiome. So basically the carbohydrate that's not being digested will go to the gut, the colon, and then a lot of bacteria maybe will have a party over there. So because a lot of food. In my lab, over more than 20 years now, we have been studying or try to understand how this very unique molecule is synthesized or produced in nature. Apparently, it's come from pseudoheptylose 7-phosphate, which is part of the pentose phosphate pathway. If you are a biochemist, presumably this is something you would be interested in. And cyclization of this molecule give 2-epi-5-epi-baleolone, which is the precursor of many pseudo-oligosaccharides. And actually, there are many, many steps involved here. I make it too short. So 20 years of work, just end up in one slide here. And then more recently, we have identified the missing pathway. So we pretty much, well, together with other investigators from other labs, we conclude all the biosynthetic pathway to these molecules. So why do bacteria produce pseudo-oligosaccharides? Coming back to the first question. So many years ago, the Pippersbeck group in Wuppertal, Germany, proposed that alkabos presumably function as carbophore for the bacteria. So it's being produced and excreted out of the bacteria. And then the bacteria also produce enzyme that hydrolyze starch. And then alkabos will react with the simple sugar and then being shipped back into the cell. So it's kind of a shuttle system that take small, simple sugar from outside the cell into the cell. But there is a competing hypothesis that presumably many bacteria, especially the one that grows slowly, would use alpha-glucosidase inhibitors as a competitive, for a competitive advantage. So obviously bacteria produce a lot of alpha-glucosidase and excrete it, and then hydrolyze starch to make simple sugar and then take it back into the cell. But the slow-growing bacteria can produce alkabos and inhibit the alpha-glucosidase enzyme from other bacteria, while its own alpha-glucosidase enzyme is resistant to alkabos, so still can do the hydrolysis, and then they can take advantage of that over the competitor bacteria. So another group of oligosaccharides called validamycin A, or I should say pseudo-oligosaccharide, is produced by Streptomyces hygroscopicus. This compound is a strong trihelase inhibitor used as an antifungal agent to treat fungal infection in rice plant. So every year, 30 to 50,000 tons is produced in China to be used for to treat rice plant. So it plays an important role in food security in Asia. But more importantly, validamycin A is a precursor of voglibose, which is a synthetic anti-diabetic drug. So it's produced by this bacteria called Streptomyces hygroscopicus. So how this compound works in the case of antifungal agents, apparently in rice plant, sometimes you have this fungal infection caused by Rhizoctonia solani. In Rhizoctonia solani, they use trehalose as a carbon source or energy supply. Trehalose consists of two units of glucose. So to get, when it's needed, then the fungi would hydrolyze trehalose to make 2 units of glucose using this enzyme called trehalase. Again, through this oxocarbenium transition state, as I mentioned just now, this half-chair form is very similar to the pharmacophore of validamycin A, which is hydrolyzed to validoxylamine A. But this CN bond cannot be hydrolyzed. So that's the way how it killed the fungi. And Takeda companies many years ago take validamycin A and then convert it to valyenamine here. So basically take this active unit or pharmacophore and then chemically modify it to make voglibose. It's known as basin. And in other countries, the names can be different. So the problem here, of course, using this bioconversion, also you can do it by chemical reaction. Sometimes you cut this bond to get polyenamine, but sometimes you also cut that bond to get validamine, which is not useful. So that's require some modification perhaps, or get different sources to get a better yield. So my lab here in Oregon State University have been also studying the biosynthesis of this molecule, validamycin A. Again, we have published a lot since 2001, it's about 20 years ago, and we pretty much know what's going on and how this molecule is produced in nature. And along the way, we also discover very interesting enzyme like this 2-epi-5-epibalilone synthase that catalyze the cyclization of pseudoheptylose 7-phosphate to make this cyclic molecule. We also found new class of enzyme that we call pseudo-glycosyl transferase enzyme that catalyze the coupling reaction of these two pseudo sugar unit to make this core molecule. And why it is important to study or to understand how these type of compounds are made. So one of them really, first you can really increase the production yield of the molecule if you know how they are made in nature. And another thing also, you can really modify the biosynthetic pathway. You can do knockout and so on. In this case, we inactivated the oxidoreductase enzyme VAL-N here. So this is the natural products. These are natural product, validoxylamine A and validamycin A. But by knocking out this gene, we are able to isolate two new compounds, one called 1,1' bis polyenamine, and then the other one, validamycin. The difference is now we have both of this pseudo sugar identical. They are identical. So when you hydrolyze this, regardless you cut the CN bond here or this CN bond, you always get the product polyenamine, which can be used to make voglibose. So this has been published and patented, and so this is some technology that one can use really to improve the natural products that nature produced for you. So we have been doing that, in many other systems as well. So because today is related to alpha diabetes, so I decided to show you on the alpha glucosidase inhibitor project that we have. So in summary, I mentioned to you just now that diabetes is on the rise worldwide, and some anti-diabetic drugs have, well, even though we have many anti-diabetic drugs, some of them have significant side effects. And then among them, alpha glucosidase inhibitors are also good anti-diabetic drug that's being used in clinics. But most natural alpha glucosidase inhibitors are produced by plant and microorganism, but I also found even marine sponges sometimes produce the molecule. Most, if not all of them, are sugar mimetics. They look like sugars. We call it pseudo sugars, but sometimes they are thiosugar, they are amino sugar or carbo sugars. Which we don't really know why they produce, presumably as a competition, but also can be a regulatory compound as well. Most, if not all of them, are produced from sugar or phosphate, either from pentose phosphate pathway, some of them even produced from glucose 6-phosphate, and so on. And biosynthetic engineering may be used to improve production and generate a new compound. Right, so I believe my time is up. So I would like to thank the people who really did the work, past and current group members and our collaborators. I would like to thank the funding agency that provide money for us to do the work. And these are the pictures that we took very recently in my backyard after more than a year being in a social distancing mode. Finally, we are now able to have gathering without worrying about wearing masks. So thank you very much. I thank you all for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Taiful. Very, very deep research you have. Yeah? So now we have 5 minutes for discussion. So, ladies and gentlemen, if you want to ask directly, I give you opportunity here. Please raise hand and you can ask to Professor Taiful Mahmud. Okay. There is no question, but I have an important question, Professor. What do you prefer for in Indonesia? Yeah, you propose to research on alpha glucosidase from plant or from microorganism?
All right. So I think that's a very good question. But if you hope from plants, the only problem with plant, of course, you require a lot of space to grow the plants, and then isolation also may be challenging, and then also modifying plants to produce more compound maybe a little bit more difficult. With bacteria, because of the genetic system of the methodology and everything is very advanced. You can do cloning, you can do gene knockout, you can do sequencing very easily nowadays. It's obviously a much better system to work with. And in fact, many people have been trying to move plant genes into yeast or bacteria to produce like artemisinin, the antimalarial drugs, or making morphine or, codeine, not only taking them from coca plants anymore, but rather just move the genes from the plants, put them into bacteria or yeast or fungi, so that you can produce them in a tank, of course, a sterilized tank, and then you can control the production very easily, and also can get the compound very quickly, within 2 weeks. You don't have to grow the plants for 10 years to get the compounds.
Okay,
so, but still, plant is a good source of natural products, but perhaps we learn what they produce, and maybe that would inspire us to produce the compound in different hosts.
Okay, but how about the marine resources?
The same thing with marine also, because especially if you get the compounds from spawns, it is not feasible to go dive, and collect tons and tons of sponges or tunicates. You will destroy the environment that way. So, and also many compounds that are produced or found in marine vertebrates, they are produced by the symbionts. They are produced by cyanobacteria or bacterial strains that live together with the animals down there.
Okay, thank you. And my short question is about, I'm sorry, it is true that alpha glucosidase inhibitors has no side effect.
That's not true. The most common side effect of alpha glucosidase inhibitors are, vomits and diarrhea and bloating. Well, most of them because of the inhibition of the enzyme that digests carbohydrates. And then the carbohydrates then will go down to colons, and that's basically digested by the bacteria that live inside the colon or the intestine, then you can produce a lot of oxygen, a lot of air, CO2 and so on. That cause side effect. And then, but some alpha glucosidase inhibitors also can be absorbed. Acarbose cannot be absorbed because it's oligosaccharide types, but like Boglibos can be absorbed. So far, no real really bad side effects coming from alpha glucosidase inhibitor. The way how to prevent that side effect also by taking it, the dose actually gradually from small amount, because it's a dose dependent. The side effect also the dose dependent. So if you take it slow and then from lower amount, and then let the microbiome in your body adjust to it, maybe then you can reduce the side effect of acarbose. But other class of compounds like sulfonylurea obviously cause a lot of problem with hypoglycemia, and also because it's induced pancreas a lot to beta cell to produce insulin. Over time, really that burn out the beta cell or our pancreas. So over time, if you use sulfonylurea for a long time, then you cannot produce more insulin. It's going down, lower and lower. And
Thank you very much. It's clear. Yes. Thank you again, so Professor Taifo. I appreciate your presentation. Thank you, and don't hesitate if you want to go to my city is Yogyakarta. So please come. Yeah. Thank you very much, Professor Taifo.
Thank you, Professor Suijio.
Yes. Okay. Thank you. Ladies and gentlemen, we now move to the last speaker for the season. He is a very famous professor from Sriwijaya University, Professor Dr. Kamaluddin. Please, organizing committee, before starting, the names is Muhammad Kamaluddin PhD, but usually we call Professor Kamaluddin. This is a famous name from Faculty of Medicine, Sriwijaya University. Please, organizing committee. Yes, he has the undergraduate program from Medical Faculty, Sriwijaya University, and then the master degree from University of Kentucky, USA, and the S3 or doctor degree from University of Pajajaran, Bandung, Indonesia. Now, the academic position really he focused on pharmacology. There are a lot of position here, and then now, and from 2005 to present, he is a professor on pharmacology, focusing in biomedical sciences and herbal medicine from the Department of Pharmacology, Medical Faculty, Sriwijaya University, Palembang. And then there are a lot of the membership of professional scientific scholarly, and then current research interest in pharmacology of neurosciences, and also on traditional medicine. It's very interesting. Please. Okay, and then there are a lot of publication, and one of his publication is very, very interesting, is a sedative effect, of and also kebugaran, it means the fatality from gaharu. This is gaharu. It's a very specific plant from Babel, from Sumatra, outside of Sumatra. So now I invite Professor Kamaluddin to speak on the I read here herbal machine for treating diabetes and other degenerative diseases. 25 minutes time is yours, Professor Kamaluddin.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you very much, Mr. Chairman, Professor Suijio Pramono, my friends. Okay, and also other participants. Now, I would like to present herbal medicine for treating diabetes and other degenerative disorders. And also, I would like to thank to the organizing committee that invite me to present. That's one of my favorites about the herbal medicine. Okay, herbal medicine is one of the very popular, not only in Indonesia but also all over the world, that have some pharmacological effect logically, and that's why we have to study everything about the herbal medicine, even though in the molecular basic and also clinical phenomenon. It's very important to make sure that herbal medicine also makes some valuable effect for our humans. But I'm sorry, in this presentation, I don't attach any clinical, any clinical results from the clinical trial in Indonesia or over the world. But I would like to show that this topic is diabetes is a chronic metabolic disease characterized by elevated levels of blood glucose. Which leads over time to serious damage to your heart, blood vessels, eyes, kidneys, and nerves. This review to say that the most common is type 2 diabetes, usually in adults, which occurs when the body becomes resistant to insulin or doesn't make enough insulin. In the past 3 decades, the prevalence of type 2 diabetes has risen dramatically in countries of all income levels. Type 1 diabetes, once known as juvenile diabetes or insulin-dependent diabetes, is a chronic condition in which the pancreas produces little or no insulin by itself. No insulin by itself. So for people living with diabetes, access to affordable treatment, including insulin, is critical to their survival. There is globally agreed target to halt the rise in diabetes and obesity by the year 2025. A need to develop safe and effective treatment modalities for diabetes because the case is increased year by year. So medicinal plants that would play an important role in the management of diabetes mellitus, especially in developing countries where resources are meager. Diabetes is a collection of metabolic disease characterized by hyperglycemia resulting from defects in insulin secretion, insulination, or both. So right now, diabetes mellitus has been divided into 4 types: insulin-dependent diabetes mellitus, non-insulin dependent diabetes mellitus, gestational diabetes, and also monogenic diabetes. According to International Diabetes Federation, there is about 415 million people have diabetes mellitus in 2015, but in 2040, it would be increased by a number of 642 million people. Now, we look at just a moment why diabetes could make imbalance between the hyper blood glucose and also to become hyperglycemia, because the metabolism of glucose is not in balanced state, like De Pranzo 1997 say that the most recommended methods to detect DM is determined by the level of blood glucose. Fasting blood glucose maybe 126 milligram per deciliter, random plasma glucose 200 milligram per deciliter, and oral glucose or stats more than 200 milligram glucose. So, which one is 10 global causes of death in 2019? About 10 cases, but 2 of them, Alzheimer's disease and other dementias, and also diabetes mellitus, is discussed today. If we look at this, the metabolism of glucose and other enzyme involving in diabetes mellitus, beginning from the pancreas and producing and releasing insulin, glucagon, and merging to the liver to become plasma glucose, tissue, and also other organs, then balancing in some manner and also in the state according to the ability of this important balancing. But after that, why glucose homeostasis as a role of insulin not so static state, but dynamic, and also give another imbalancings by the time, because some of factors here, from the pancreas, liver, and also other organs, also influence the blood glucose in balance. So we have to ask about why should we choose plants instead of conventional medicines. The usual reasons, because its effectiveness relatively safe, for fewer side effects, and also affordable and low cost, especially in tropical countries such as Indonesia. So the herbal medicines and also other herbals available can be managed some cases of DM should be explored in order to make a cure for the DM in the future. The main active constituents of plants for treating diabetes type 2 include alkaloids, glycosides, galactomannan gum, polysaccharides, and also peptidoglycan, hypoglycans, guanidine, steroids, carbohydrates, glycopeptides, terpenoids, amino acids, and also inorganic ions. Now, some plants could significantly regulate well organs involved in such a disease, such as the pancreas, liver, muscle, and adipose tissue. On the other hand, pancreatic beta islet are responsible for insulin production to be augmented the islet size, cell size, and the number of insulin secretion level. So the liver, the most crucial organ to sense fluctuation in blood glucose concentration, and could be responded via buffering mechanism accordingly. Some plants involved in several key enzymes, phosphoproteokinase, phosphorylase, glucose 6 phosphatase, etc. Transporters, GLUT2, GLUT4, etc., as well as cell surface receptors, ISTHM, and others. In hepatic, muscular, and adipose tissues are major targets for anti-diabetic drugs. And also, such control of blood glucose levels can be via insulin-dependent protein kinase B or insulin-dependent AMP, activated protein kinase signaling cascade. Several drugs target to soothe the cell damage associated with hyperglycemia. Say antioxidants have invaluable effects that shouldn't be ignored. And the simple routes of glycemia controlled by phytochemical drugs are via the inhibition of glucose digestion and absorption. So the main active constituents of these plants include alkaloid glycosides, galactomannan gum, polysaccharides, peptidoglycan, hypoglycans, and guanidine, steroid carbohydrates, glycopeptides, terpenoid, amino acid, and inorganic ions. So this affect various metabolic acids, which are indirectly affect the level of blood glucose in the human body. For example, the important biochemical pathways where blood glucose is involved, glycolysis and Krebs cycle, inhibit gluconeogenesis, especially glucose 6 phosphatase, synthesis of glycogen by affecting the enzyme glycogen synthesis action, regulate glycogenolysis by regulating glycogen phosphorylase, and also interfere digestion and absorption of carbohydrates by inhibiting glucose oxidase activity, TAGOG activity, and also enhancing glucose transporters, and potentiates insulin action by inactivating insulin enzyme. Here we can see how glucose are managed by our body with any enzyme here, hexokinase, sulfonylurea, and also calcium channel open, or make a depolarization, and then glucose and other important such as insulin and etc. could be managed in our body. And so here insulin with the IS1, and also GLUT for translocation, can also make it balance glucogenesis, beta cell growth, insulin secretion, glucose transport, and also protein synthesis, and other gluconeogenesis, glycogenolysis. Here we can see insulin-dependent and independent regulation of glucosidic and lipids metabolism by three organs, important organs of our body: fat, tissue, liver, and skeletal muscle. And also, how would be in dangers because the glucose is not regulated well. So here, kidney lens intestine by fructose orbital, nerve lens, and also other osmotic damage would be cataract formation and glycosylation, lens of eye. And the other, glucose 6-phosphate, will manage glycogen, kidneys, glycosid, kidney capillaries, and defective nutrition and transport. Here are some of our herbal medicines, such as Anoectochilus roxburghi will regulate beta cell function. Capsicum plants also regulation of insulin resistance and probably beta cells. Curcuma longa regulation of insulin resistance.
Saya minta maaf tidak ikut pesta.
Cells.
Tidak pesta.
And curcuma longa and also zingiber officinalis also regulate.
Karena saya tinggal.
And then diabetes is metabolic disorder, alarming health problem, and we have to at least
itu concern saya kan itu yang
explore again 802 plants having hypoglycemic effects as anti-diabetic plants prospectively for future treatment of diabetes mellitus remedies. This is the other popular medicinal plants for diabetes reported by participants and also by doctor, according to Al Jawara 2020. Yeah, we have here cinnamon, ginger, other fenugreek, black seeds, fennel, garlic, turmeric, onion, basil, and colocynth. We look at this one. It's between some medicines and also doctors look like the same idea, the same interesting observation here. And also another plant, angel, mamus, alum, chipa, alum satiponel, etc., should be explored in the coming year because mode of action is look like a logic for the pathogenesis and pathophysiology of diabetes mellitus. Now, we also talk about the neuroprotective potential. There are about three main categories: natural products with neuroprotective potentials, toxicology studies of natural products and hepatotoxicity, and effect of natural products on oxidative stress-related disease, and effect of natural products on endocrine disease, metabolic syndrome, and osteoporosis. Yeah, we could list some of cells in aging process becoming older, get some degenerative even in cells and also another tissue and also vascular, and this is cells typically for Alzheimer's disease and dementia, spinal muscular atrophy, Huntington disease, Lewy body dementia, and Parkinson disease. Most degenerative disorders have no cure, but medication and treatment can help improve symptoms, relieve pain, and improve mobility. Here, we look at this low enzyme output A, and so became resveratrol, reporting by Murtaza. Seems that it radix ginseng, colis, katsuri, herba, abri, and also valium, elysis currently look like can interfere the pathobiology and also pathophysiology of Alzheimer's disease. In the normal brain, we see that the cortex still takes, but in the Alzheimer's brain, it's very thin. And also another organs in our brain look like very, very shrinkage, especially in the hippocampus. So Alzheimer's disease, AD, Parkinson's disease, and also most common neurodegenerative disease, usually start slowly and progressively worsen. It is caused 60 to 70% of cases of dementia. And the most common early symptom is difficult to remember recent agents. Other degenerative disease affecting circulatory system and also neoplastic disease may be get cancerous disease. So Alzheimer's disease is a progressive neurodegenerative disorder of the brain, which leads to a decline in memory and cognition. The Food and Drug Administration, approved administration, provide only symptomatic relief and are often associated with adverse effects. Many natural products have shown their activities against AD, but bioactive compounds like polyphenol, tannins, alkaloids, flavonoids, terpenes, sterols, etc. are present in various plants. These compounds have shown antioxidant, anti-inflammatory, anticholinesterase, and anti-amyelogenic activities. So AD characterized by neuronal dysfunction often associated with atrophy of the affected nervous system structures. Age-related dementia constitutes a major subset of neurodegenerative disease, and also AD is the most prevalent clinical form of dementia in the aging population. 43% of individuals over 85 years of age are thought to have AD in the United States. Parkinson's disease may be another prevalent neurodegenerative disease among the elderly, affect 1 till 3% of the population over 60 years of age. Kim reported effects of a mechanism of 6 general gingerol, potent ingredients of ginger, on a scopolamine-induced dementia. And also Liu demonstrated anti-amyloid precursor protein APP cleavage and amyloid beta defects of human quinone decoction extract, especially extract in China, as a Chinese herbal formula on the special learning and memorability to transgenic APP PS1 mice. So I come to my conclusion. Herbal medicine shows mechanism of action can interfere pathogenesis and pathobiology, DM. So this finding would be investigated clinically and also molecularly to imbalance factors involved in diabetes mellitus disease. And herbal medicines also interfere pathobiology and imbalance molecular factors in neurodegenerative disease. So it would open view for doing research in the future. So that's all my presentation. I'm sorry that I cannot support at the time the result of a clinical trial in the Alzheimer's disease and also in the EM. Thank you, Mr. Chairman.
Okay, thank you, Prof. Kamaluddin. It's very deep. Really a lot of article here, and then more clear about the diabetes. So we have several minutes only for the discussion. If you have a question, please raise your hand, and then I can deliver to Professor Kamaluddin if you have. If not, I have a small question, Professor Kamaluddin. In the present moment, about the okay, there is a raised hand. No, resistance insulin, and then deficiency insulin. What one you prefer to make the research in the present moment for traditional medicine in Indonesia or medicinal plan of diabetic in Indonesia?
Thank you for your question. I think the resistance of insulin is very important to be explored and also investigated. Why in Indonesians much more insulin insistent cases instead of the reduce of production of insulin. Until now, some investigators said because the Indonesian people not like to use of this herbal medicines more and only use the herbal medicines as a vitamin, also other energy inducers, and that's all. So I think it would be better because Indonesian people should memorize and also understand why insulin is instead of reducing insulin production, got some more cases in Indonesia.
Okay.
Yeah.
Well, thank you. I agree with you. So this is more challenge for us, yeah, to do the research on medicinal plant in Indonesia for the insulin resistance. Okay, ladies and gentlemen, time is up. So if you have question, etc., you can ask to the organizing committee and then can deliver to both Professor Kamaluddin and also Professor Taifu Mahmud. So as moderator, I thanks Professor Kamaluddin and also Professor Taifu Mahmud. I back to the organizing committee. If, for example, I have some mistaken in the as moderator, I apologize. So thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Warahmatullahi wabarakatuh. Thank you very much, Professor Swijio Pramono, for guiding us in the first session. And also, I would like to thanks to all of the plenary speakers. All the certificate and souvenir from us will be sent directly in the home address. Next session, there will be three keynote speakers presentation that will be guided by Professor Sutopo Hadi. But before that, let me read his brief curriculum vitae.
Terima kasih banyak kepada Profesor Swijio Pramono yang telah memandu kami di sesi pertama dan seluruh pembicara pleno. Semua sertifikat dan souvenir dari kami akan dikirim secara langsung ke alamat rumah masing-masing. Sesi selanjutnya akan dipandu oleh Profesor Sutopo Hadi. Sebelum itu, izinkan rekan saya untuk membacakan kurikulum vitae singkatnya.
Professor Dr. Sutopo Hadi MSc is a professor at the Department of Chemistry, Faculty of Mathematics and Natural Sciences, University of Lampung. Here are his education background. In a bachelor, he graduated from Lampung University in the field of study synthetic organic chemistry. In the master degree, he graduated from University of Queensland, Brisbane, Australia, with the field of study organometallic chemistry, and also in the doctorate degree in the same university in University of Queensland, Brisbane, Australia, in the field of study bioorganometallic chemistry. Next, there are peer-review publication of Professor Sutopo that I will read some of them. In 2021, synthetic and comparative study on the antibacterial activity organotin 3-hydrobenzone compounds in the field of study pure and applied chemistry. And in 2021, analysis of mercury in the skin lightening cream by microwave plasma atomic emission spectroscopy in the field in the molecules. Next, for book writing in 2030, Professor Sutopo made the organotin chemistry synthesis, characterization, and application. In 2012, General Chemistry 1. And then in 2007, analysis and characterization with AR and UV vis spectroscopies and centrifugal activity tests of dibutyl and dipenyltin carboxylate. And for patents, in 2030, 3-penyltin benzoate compounds as a new anti-cancer agent, and in 2017, 3-penyltin nitrobenzone compounds as a new anti-corrosion inhibitor. And the last, for awards in 2030, as the University Outstanding of the Year by University of Lampung. In 2012, elected as one of expert editor in a book and journal writing at DHERR by Directorate of Higher Education Ministry of Cultural and Education, Indonesian Republic. So, ladies and gentlemen, please welcome our second moderator, Professor Sutopo Hadi.
Mari kita sambut moderator kedua kita, Profesor Sutopo Hadi.
Thank you. Thank you, the Miss and Mr. Master of Ceremony. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And good afternoon, everyone. the participant of the ISTHM seminar 2021. I'm honored and very happy to be here with all of you to be a moderator for the second session. In this session, we will have three great speakers. The first one will be Professor Dr. Asep Sukohar MSc, the Medical Doctor Association Head of Lampung Branch. And then the second keynote speaker will be Professor Dr. Kerry Lestari Dandan, MSc Apotheker from the Faculty of Medicine University of Bajajaran. And then the third keynote speaker will be Professor Dr. Engineer Yuli Widyasuti MP from Pekcanas TOT. Now let me read the curriculum vitae of the first speaker, Professor Dr. Asep Sukohar. Currently, he is the Vice Rector of Finance Affairs, General and Finance Affairs of the University of Lampung, 2020-2022. He is affiliated with the Faculty of Medicine, University of Lampung. Next. Wait. Hey. He is graduated the undergraduate from the Faculty of Medicine, University of Trisakti Jakarta in 1995, and then the Magister from the Medical Faculty of Pajajaran University, and then the PhD, the doctoral degree from the Pajajaran University. Now, to short the time, I would like to remind you, Professor Asep, we have only about 25 minutes for you to give us a lecture. And then because today it will be parallel session, maybe the audience later on will ask the question after all three speakers finish the presentation. Now time is all yours, Professor Asep Sukohar.
Thank you, moderator, for my time. Maybe please allow me to share my presentation by myself. Please allow me. I will share my presentation. Okay, thank you. Thank you very much for moderator for your time, and this is my time. I will first call about Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera for all the participants and tabipun. And then this is my presentation. Is relative relation with the traditional medicine and herbal medicine and degenerative disease. And this is my research. We develop about the traditional medicine from natural, and that is we extraction and isolation and purification, and we have done about the research in the laboratorium since 2010 until now. And this is my presentation about the Kurkosun B from Jatrofa and Jatrofa Kurkas. We began. This is anti-proliferative activity Jatropon and Kurkosun B from Jatrofa Kurkas on hepatocellular carcinoma PLC5 cell lines. Okay, we continue. We know that this cancer has been a serious health problem over the world when continuously increasing prevalence every year. International Agency for Research of Cancer in 2018 In 2018, reported 18.1 million new cancer sufferers with mortality rate 9.6 million. Hepatocellular carcinoma were the sixth highest prevalence cancer and the fourth of the cancer-related death, with dominated by the Asian race. World Health Organization was estimated more than 1 million patients will die caused by liver cancer in 2030. Wait a minute. The prevalence of cancer in Indonesia also reported increases from 1.4 per 1,000 population to 1.8 per 1,000 population in 5 years. Hepatocellular carcinoma is the second high prevalence cancer by 12.4 per 100,000 population and mortality rate 7.6 per 100 population in the world. The development in herbal medicine or traditional medicine and technology on hepatocellular carcinoma treated was increasing rapidly. This is my research we have done since 2010 to answer about the cause of the treatment cancer, and we develop with the herbal medicine with low cost. That is my my destination. However, there are no clinical satisfactory therapeutic results. The current management still have several issues about side effects and high cost of therapy modalities. For many years, traditional herb medicine have been used as medical treatment, especially in developing countries like in Indonesia. There are about 40,000 plants in the world, and third reported in Indonesia. That is more than 940 have utilized as traditional medicine. Herbal medicine are being considered as a possibility strategy against on hepatocellular carcinoma. Jatropha curcas is multiple-purpose plant with some potential medicine use. Number of study was conducted to analyze medicinal properties compound, phytochemistry, and its pharmacology as in as anti-cancer. We know that Jatropha is rich of terpenoid as its activity compound, and some terpenoid compound from Jatropha such as Jatropha, spirosenol, Jatrotrion have exhibiting effect as anti-tumor against in laboratorium to lymphocytic leukemia. Curcosunbi exhibiting effect as anti-invasive against cholangiocarcinoma cell. Some studies have conducted to analyze curcosunbi as anti-cancer. That we learn about the cholangiocarcinoma cell and chronic myelogenous leukemia cell line and human non-small cancer, lung cancer, and human epidermal carcinoma cell line, and human hepatocellular carcinoma cell line, and human breast adenocarcinoma cell lines, and the other cell line. That is, we have learned in journal. And another studies to assess its cytotoxic effect that we can compare with the other cell line. The method, the study was conducted on March 2016 until May 2017. 17 at the Research and Development Department of Darmais Cancer Hospital in Jakarta. That is hepatocellular carcinoma with type of VLC5 cell line where culture used the MEM medium from YIPCO, and cells were harvested after 80% confluent with trypsination method and planted with 10 quadrat 5 cell line per well in 1996 well plate. Cells were incubated with again using the MEM, and cells are ready to be treated after the medium with FBS discard. The calculation of IC50, that is Jatropon and Kurkoson B, was calculated by Fine, Patton, and Whale, and Lissit, Wilcoxon, and Miller and Trinton. The presentation of that cell result by MTTSA will transform it to probit value or probability versus log concentration. We compare that is presentation of that cell count by the formula 100 minus percentage of viable cells. And we can look at in this figure, we look at the Kurkosun B concentration. We look at 1 until 5. This is the decrease of the concentration of the Jatropon. And we look at this viable percentage of viable cells. This is we can look at for this. This is the increase. We look at on the log dose. This is the decrease of value. We can look at in this figure. This is not is like line, but we can look at this is the line in we compare with the media. This is different with the Jatropon. If Jatropon we compare with the Kurkosun B, this is alignment is smart. It's like line. I think this is more better if we compare with the Kurkosun B. Okay. By the interpolation graphic of Kurkosun and Jatropon, that is log IC50 formula. We compare A for the Kurkosun B and B for Jatropa. In R quadrat is more or less same. By using this calculation, it was known that IC50 of Kurkosun B and Jatropon value were 9.57 micromolaritas and 8 and for Jatropon 8.78 micromolaritas respectively to inhibit to activities of hepatocellular carcinoma with type of VLC5 cell lines. Based on NCI USA criteria, the IC50 of Kurkosun B and Jatropon in this study have activities to be developed as anti-cancer modalities. The result of the study, that is, we can analyze Kurkosun B as anti-cancer agent. And we compare some value in the journal about IC50, Kurkosun B to cholangiocarcinoma, chronic myelogenous leukemia cell line, and human non-cell lung carcinoma cell line, and human epidermal carcinoma. We compare about the value of micromolaritas. That is Kurkosun B with hepatocellular carcinoma type of VLC5. That is better value for category anti-cancer. But this research only on laboratory or not in human. That is, we need for the next study in clinical trial. And then cancer cell line and Jatropha necrosin B has also been studied again to type of human cell line among cervical cancer cell line HeLa and colon cancer cell line. That is the value 5.13 micromolaritas and 18.25 micromolaritas. Our study of use of Jatropon in another cancer cell line in HepG2 1886 cell line, we obtain about 3.2 micromolar. This is the same with our study in Dharma S2. Hepatocellular carcinoma therapies, as we compare with cidofovir and ribavirin, now have IC50 by 24 micromolaritas and 86 micromolaritas. In addition, sorafenib, drug of sorafenib and OSU2S, the latest anti-cancer drug, as hepatocellular carcinoma versi 5 cell lines therapy have IC50 by 9.63 micromolaritas and 8.96 micromolaritas. As the result, the cytotoxic activity of Jatropon and Curcosun B have a better effect against hepatocellular carcinoma with type VLC5 cell line when compared to the other human cell line. The mechanism of Jatropon and Curcosun B on inhibition of cancer cell growth by molecular mechanism has not been clear until now. However, some studies guess the mechanism were associated by inhibition of arachidonate and of arachidonate acid metabolism. The metabolic product of arachidonic acid we call COX and LOX pathway have shown play an important roles in various physiological and pathological processes. In malignant cell, arachidonate acid have been overexpressed and involved in multiple stages of carcinogenesis by modulation, xenobiotic metabolism, apoptosis, immune response, angiogenesis, and invasion and metastasis. However, the molecular target and mechanism by which Jatropon and Curcosun B work on cancer cell are being further explored. The conclusion that Jatropon and Curcosun B have shown potential as anti-cancer agent. It has better cytotoxic activity against hepatocellular carcinoma with type VLC5 cell lines compared to another human cancer cell lines. In addition, Jatropon and Curcosun B also have better anti-cancer activity on hepatocellular carcinoma with type VLC5 cell lines compared to anti-cancer drugs such sorafenib, cidofovir, and ribavirin, which has used widely. But this is we compare in laboratorium and in journal or meta-analysis. Thank you for the time, the moderator, Prof. Topo, and we give you time again.
Thank you, Professor Asep Sukohar, for the wise time used in this session. please stand in the Zoom meeting because we will listen to another two keynote speaker, and then after that, maybe participant will have some question to either to the keynote speaker later on. Now, let us read the second keynote speaker for this session. Will be Professor Dr. Keril Lestari Dandan from the Pajajaran University. Professor Dr. Abtekar Keril Lestari graduated from the pharmacy of University of Pajajaran, and then apotheker also from Pajajaran University, magister from the pharmacology ITB, and then a doctoral degree from the pharmacy of University of Pajajaran. There are so many organizations she has included in, and then now she is the director utama of in Jabar University of Pajajaran. There are so many awards that she has received during her activities at the Pajajaran University. Now, to save the time, let us welcome Professor Keril Lestari to give keynote speech. Would like to remind you that we will have only about 25 minutes, Professor Keril. Time is now all yours.
Yeah. Thank you, Professor Sutopo, and then also thank you for the committee to arrange this seminar. Very good. And then allow me to share the screen.
Oh, Prof, please unmute your speaker. Hello, Prof. Please unmute your speaker. We haven't listened to your voice. Professor Kerry, could you please unmute your speaker? Thank you.
Okay, thank you. I'm sorry. Okay, a very good day for all the participants. Thank you for Professor Sutopo, and then for all the committee who arranged this seminar, international seminar. It is very inspirative event for this pandemic, during this pandemic COVID-19. So my the title of presentation is Stevia tea as a functional beverage to improve blood glucose control as a downstream herbal research to market. I'm Keril Lestari from the Department of Pharmacology and Clinical Pharmacy, Faculty of Pharmacy, Universitas Pajajaran. So maybe this is also, I'm not from the medical faculty, but I'm from the Pharmacy Faculty of Universitas Pajajaran, and also now I as a commission in National Commission for Jamu Scientification, Ministry of Health, Republic Indonesia. Ladies and gentlemen, all the participants, as we know, the prevalence of diabetes mellitus is growing every year, including in Indonesia. Although many of drug is exists for enhance for the therapy of diabetics, but still the performance of therapy is not to compete all the purposing of the therapy. So the research on medicinal plant or medicinal herb were used to lowering the blood glucose is still ongoing, and then hope we have the new therapy for enhance the therapy of diabetes. One of the potential help to have hypoglycemic activity is Stevia rebaudiana bertoni. We know Stevia rebaudiana bertoni leaves is content of the active compound as steviocyte, and then also as we know that stevia is a steviocyte is a low calorie sweetener with sweeter than just like other sweetener, and then the stevia have 300 times sweeter than sucrose. And as we know that in type 2 diabetes mellitus, the metabolic disorders is punctuated by the insulin resistance. In type 2 diabetes mellitus, the resistance to insulin is one of our concern. The factor may be triggered by the high consume of calorie intake, and also obesity and sedentary lifestyle. As we do on work from home, we still goes to the sedentary lifestyle, and also as well as a genetic. And in epidemiological research in developing country showed that due to the high older population structure and high rates of obesity, the incidence of prevalence of type 2 diabetes mellitus still increasing. And according to the World Health Organization, Indonesia has the fourth largest diabetic population in the world after India, China, and the United States. So due to this very huge population, the research on the diabetic disorder and also the hypoglycemic agent is still very potential to develop. And one of our concern is Stevia rebaudiana bertoni. Stevia extract. The extract of Stevia rebaudiana bertoni as a hibiscus plant has been used for many years in the treatment of diabetes among the indigenous people in Paraguay and Brazil. Stevia rebaudiana bertoni leaves contain steviocyte, a natural low calorie sweetener that 300 times sweeter than sucrose with 0 calorie, as we know. And a number of recent studies revealed that consumption of the stevia can lower the plasma glucose level. This is very advantageous for the treatment of the diabetes. Why? The diabetician still can taste the sweet things, but these sweet things is also can manage the glucose level. And then extract of Stevia rebaudiana bertoni can stimulate insulin secretion, and then also stimulate also the sensitivity or the sensitivity of the insulin by acting directly on pancreatic beta cells and showed antioxidant properties. Steviocyte and steviol that extracted from the Stevia rebaudiana bertoni show to increase expression of glucose transporter type 4 or GLUT4, and also GLUT4 gene and protein, and also glucose uptake in cell. With this mechanism of action, one of our systematic reviews showed that not just as an anti-diabetic, but also Stevia rebaudiana leaves also can act as also have the activity as the antioxidant and anti-inflammatory, also anti-hypertensive, also anti-hyperlipidemic, anti-obesity, anti-cancer, antimicrobial, antiviral, and effect on the renal function and effect of the kidney function. This effect is very advantageous or very promising to develop the stevia, but in this research, we just concern on the anti-diabetic, or our concentration in anti-diabetic. The study of the Stevia rebaudiana leaves as an extract as a nutraceutical with hypoglycemic activity in diabetic rats is already published in international journal Scopus Index. A study was conducted to examine the hypoglycemic activity of stevia leaves extract in diabetic rats. We found that the average of fasting blood glucose level in induction of the type 2 diabetes mellitus like this. So we use the male Wistar rats were feed high fat and high carbohydrate feed and sugar solution for 74 days to induce the diabetic rat models. This is the methodology to induce the diabetic rat models in type 2 diabetes mellitus. The animals were then divided into 5 groups consisting of a negative control group treated with 2% pulviscum arabicum and a positive control group treated by metformin 45 milligram per kilogram body weight, and the 3 test group treated by aqueous extract of the stevia rebuna bertani leaves at any dose like we see on the presentation. And then they treat for 36 days, and the blood glucose were measured on the day 14, 28, and also 36. This is the numbers of blood glucose level before we do the intervention. And then after we do the intervention, this is the number of average glucose level fasting rats graph during 36 days of the treatment. As a result, show that blood glucose level over 36 days after we use the intervention were significantly lower in the group of treated stevia rebaudiana bertani leaves extract, and then also and then also we analyze with the further human culls. We analyze further with human culls suggested that hypoglycemic activity of stevia rebaudiana bertani leaves extract was dose dependent. So we try to find the best dose to decrease the glucose level of the glucose in the drug, glucose in the drug. And then the conclusion is the result indicate the stevia rebaudiana bertani leaves extract has a potential role as a hypoglycemic agent in the treatment of type 2 diabetes mellitus. And then for the mechanism of action, or the several mechanism of action for the stevia leaf extract as a hypoglycemic agent is shown by several research follow this research, like from Carrera et al. From 2016 said that the stevia rebaudiana lowers blood glucose level by increasing insulin secretion and decreasing glucagon secretion in alpha pancreatic cells. The other way, Carrera also said that on 2017 also said that stevia, which is the major compound of stevia side with the major compound of the stevia rebaudiana bertani, has the effect of stimulating insulin secretion and increasing insulin sensitivity by acting directly on pancreatic beta cell. And the other research on 2016 said that the stevia rebaudiana bertani extract increased the expression of peroxisome peripheral activated receptor gamma or PPAR gamma and insulin mRNA. PPAR gamma plays an important role in maintaining glucose metabolism by stimulating glucokinase and glucose transporter 4 in the pancreas and liver. By increasing the expression of PPAR gamma and GLUT4, stevia will enhance the glucose uptake into the cell. Thereby, the glucose in the blood were metabolized and resulting the lowering blood glucose level. Ladies and gentlemen, now we try to we try to downstream this research to become a product to market. And then fortunately, we already has launched the new product to market on 2018. The name of the brand is Tehdia. Tehdia is one of the tea with stevia, and then Tehdia do as a drink, and then also we can use as a functional drink for health in the form of the sweet tea bags without sugar. So we can also have the tea without sugar, but already have the sweet taste. Yeah, the Tehdia made of a mixture of the black tea leaves and stevia leaves. The previously discussion that stevia leaves extract is good for the body, and then especially for the diabetes, because it can lowering blood sugar levels, but it also have the sweet taste. So the Tehdia can be used as an option, as a sweet drink that can be consumed daily without worrying about the increased blood sugar levels. But some obstacles is rise when we do formulate the black tea and the stevia leaves. The obstacles is the aftertaste of the stevia leaves. But after we use formulation technique from the pharmaceutical way, we can enhance the performance of the formulation, and now the obstacle of stevia aftertaste already well managed. And then this is the overview about the product, the downstream of the stevia leaf as a product to the market.
Tehdia, created from a long process, with natural sweetness taste in every cup. Produce a real collaboration and cooperation in maintaining the authenticity of Tehdia. UNPAD is one of them. This university has involved directly in developing Tehdia. Ensure everything precise. Reiterate to make sure everything according to standard. Wholeheartedly picked by the hands of the farmer who are involved directly. Stevia planted with the best seed for perfect result. With an organized process. This is the product of hard work, until it finally perfected to preserve the quality. The best performance from all the aspects prepared, with precision in all of the necessary aspects to produce and process Tehdia. All the steps according to the standard from the stevia mills. Mixing stevia and tea. Enclosing Tehdia. Testing. Until packaging and keeping Tehdia in the storage room. Tehdia becomes the natural guide of stevia and tea. Tehdia, the pleasure to enjoy your tea without any limitation, without fear of diabetes.
Demikian Bapak Ibu. Dan sekarang ini, and now, also we have already developed the 2 variant of Tehdia, and then also already patented in collaboration with PT DPE Indonesia. This is the inclusive program. This is not just as a tea or a herbal tea, but also there is some farmer has a doing in the ecosystem of the business. So also this product is already registered BPOM by and also have the halal certified and HACCP certified. With the HACCP certified, now with the we try to enter the export in the USA market, and then we already has the collaboration with the Bozeman Global in Georgia, USA. Now in progress to FDA. FDA registration, and then this is the nutrition fact of the stevia tea. There is the numbers of the fat, cholesterol, and also the calorie is 0. And then we are also proud because this is the 100% product of Indonesia will be goes to abroad as an export product. And then this is the testimony about our from the doctor about our product.
Well, karena kami kan yang seperti saya bilang udah meneliti herbal, udah tahu gimana efeknya itu sangat hebat kalau sebenarnya digunakan dengan tepat gitu kan. Kalau seperti herbal itu. Jadi ketika mengetahui kayak produk herbal seperti daun stevia pada teh dia itu, orang tua saya langsung percaya dan mau nyoba karena kami tahu kayak mana efek herbal itu kalau diproses dengan benar gitu. Karena udah mau diabetes jadi minum obat untuk menurunkan gulanya gitu. Hampir 10 tahun, tapi semenjak minum teh dia ini jadi udah stop minum obat. Dia udah sakit gula tapi masih suka minum teh, udah pakai teh dia aja. Setidaknya kok memang masih hobi minum teh, nggak yang membahayakan gitu. Ini aja aman. Jadi hobi dia minum-minum yang sesuatu yang manis tetap ada, tapi tetap aman untuk tubuh dia.
Yeah, there is the testimony of our product, and then this is the reference. Thank you so much about your also about your attending of our presentation, and then hopefully this is one of the product of Indonesia that it becomes a downstream and then goes to the market and also can be useful for the human being and then can be useful for the community healthy. Thank you very much.
Thank you, Professor Carol Story, for the very wise time of use in this presentation. Please stand in the Zoom meeting because after the presentation of the third keynote speaker, there will be maybe some question from the participant. And I will conclude later on all together for the three keynote speaker. Now let us listen to the third keynote speaker in this session, which will be presented by Professor Dr. Engineer Yuli Widyasti MP. She is now senior researcher in the P2P2TOOT, yeah, the Center for Research and Development of Medicinal Plant and Traditional Medicine. Professor Yuli, the next slide please. Next. She graduated bachelor degree in agriculture from 11 Maret University in Solo, master degree from agronomic department 11 Maret University, and then the doctoral degree, Doctor of Biotechnology from Gajah Mada University in 2017. There are so many membership she has involved so far, and then lastly, she is a member of the Health Research Ethics Committee from 2021 until now, and others also still active for all the membership of professional societies. Now, to save the time, Professor Yuli, I would like to remind you that you have only 25 minutes to present your talk, and then after that we will have a discussion with the audience. Now time all is yours, Professor Yuli.
Okay, thank you, Professor Sutopo, for your nice introduction. So let me share my presentation.
Professor Yuli, could you please switch on your camera?
I already Yeah, I already switched on my camera. Okay. Yes. Distinguished participants, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. First of all, I would like to thank to the organizing committee for having me to this important seminar today. I will talk about ADAS, Funiculum vulgare, and today's presentation actually is one of the mandatory from the National Working Group of Medicinal Plant and Traditional Medicine to review all the result aspect from ADAS. And this review will be compiled as a book series from National Working Group of Medicinal Plant and Traditional Medicine. And previously, we already published 4 books. First, Pegagan or Centella asiatica. Secondly, we already published Cambiloto or Andrographis paniculata, and Sledri or Apium graveolens, and the last one is a series or Piper better, and this species will follow the previous published book. Ladies and gentlemen, as we know that the ADAS of Funiculum vulgare already the research of ADAS has been conducted all over the world, and we easily found the information in scientific database from such as from Google Scholar, from Science Direct, Scopus Link, PubMed, and so on. Based on the keywords of Funiculum vulgare, we can get 44,000 information regarding the ADAS research from Google Scholar, 149 results from PubMed, and 2,404 from Science Direct. So there are so many research already published in the scientific database worldwide. So my talk will include botany and cultivation of fennel or Funiculum vulgare, post-harvest handling, phytochemistry, pharmacology, and economy benefit as well. In order to provide the comprehensive information regarding to the final research from botanical aspect until the economic prospect. Fennel or ADAS in Bahasa, we know that this is a kind of medicinal plant used since immemorial time. In Java, we know that ADAS very familiar, knowing as Adas Pulawaras, because the Adas always combined with the Pulau Sari bark. It is very famous in Java, and from the ethnics, Java ethnics, the Adas Pulawaras, it is used for any kind of jamu formula. We can find this formula in jamu godokan. It is the boiled jamu in Java, and this is still remain the questions because Java ethnics Java ethnics try to combine two kind of medicinal plant to be one formula, such as kunyit asam, turmeric with turmeric, Javanese turmeric, and beras kencur. It is combined for adas, rice and galangas, and also the kudu laos or mengkudu. Mengkudu, it is one of very famous medicinal plant in Java also, and laos or galangas. And adas Pulau Varas maybe has specific properties, medicinal properties that still to be explored in the future time. And fennel, it is naturalized crop in Indonesia. It has come from Europe, and it has been cultivated in many Indonesian areas, especially in the mountains regions, because it only grow well in the high altitude, more than 1,000 meters above sea level. If we take a look to the history of this plant, fennel was well known in the ancient world and very popular in the Roman Empire. Fennel not only used for medication, but it is used for a spice. It is have a very flavoring agent refer to the essential content within the seeds, and the leaves also used as a popular vegetable, not only in Europe but also in Indonesia. Funiculum is a Roman name, came from the Latin word foenum or hay, and was corrupted to funculum in the Middle Age, where the common name fenkel comes from maybe. So it is very popular with the vernacular name of fennel in the international trading. The botany of Funiculum vulgare, the species name is Funiculum vulgare Mill, or Anetum funiculum, the synonym, and Funiculum capillaceum or Funiculum officinale. The miller is come from the Mr. Philip Miller. It is accredited to Mr. Philip Miller, who first validity published in the 8th edition of Gardener's Dictionary in 1768. From the on, the name of this plant written as a Funiculum vulgare. The taxonomy of Funiculum vulgare, it is belong to the Apiaceae family. This is similar to other Apiaceae families such as ketumbar or coriander, carrot or wortel, and so on. So why these plants have a specific aroma refer to the chemotaxonomy, because this come from the Apiaceae family, indicated that this family have an essential oil content in the plants. The description of the Funiculum vulgare referred to the publication from Purwaningsih and Brings. It is very detailed description of this plant. It is sometimes annual, perennial, or biennial, depends on the region which where these plants grow. This is the vegetative plant, and this is the flowering plant, and this is the seed. This is the dry seed, which is common found in the market like this, and this is the stem. Actually, it is a basil stem. This is used for vegetable also in Europe, but this kind of stem we cannot find in Indonesia easily because this is another variety of Funiculum vulgare. Vernacular name refer to this vernacular name. We can find almost 100 vernacular name around the world because Funiculum vulgare or fennel have been distributed all over the globe, from Europe to Africa to India to Asia until to South and North America. In Indonesia, we known by several local name. Adas in general, we can found easily in the market. Adas Londo or Adas Londo because we have also another Adas such as Adas Anis or Adas Pahit refer to the other species. Adas Anis comes from Anetum graveolens and the Adas Daun come from the other species of Rutaceae family, and Hades for Sundanese name. The origin and distribution of the fennel, this plant actually originated in the southern Mediterranean region, but it has been naturalized around the globe, including Indonesia. In Indonesia, fennel is cultivated all naturally in the mountainous area. As I mentioned before, the fennel could be found in the Boyolali in Central Java, or in Bukit Tinggi at Sumatra, or in other areas with the elevation more than 1,000 meters above sea level. It has been cultivated and also grow wild throughout the northern, eastern, and western hemisphere, especially for Asia, North America, Africa, and Europe. The genetic research revealed that Funiculum is diploid, having 2N equal to 22 chromosomes, with large number of varieties and races. Two distinct subspecies are recognized, Piperetum and Capillaceum. And Capillaceum has 3 varieties, Azoricum, Dulce, and Vulgare. And 2 other varieties, Azoricum and Dulce, are not found in Indonesia. We only have Vulgare varieties in Indonesia. This is the figure of fennel varieties. This is varieties Azoricum, Dulce, and this Vulgare. In Indonesia, we easily found the Vulgare varieties only. The cultivation of fennel have been intensified, carried out in India and China. The 2 countries are the main producer of fennel in the world. There are so many research have been conducted to increase the production as well as the essential oil content of fennel around the world also, but unfortunately it's very limited found in Indonesia. So maybe it is still open to researchers in Indonesia to do the research according to cultivation of fennel to increase the production of fennel seed. To get the proper quality and proper quantity of fennel seed, it should be cultivated in the particular environment condition. This plant need a dry and cold weather in a long sunny days, but Actually, this plant tolerate with all types of soil, which is rich in plant nutrient and lime, and pH range from 6.5 until 9, until 8 is preferable. And planting date early rainy season, October or November in tropical region, but mainly in the subtropical region, it's only could be grow in summer season. Cropping system refer to the farmer in Indonesia. Sometimes it is grow monoculture. It means in one land it is only cultivate the fennel, and also grow as an intercropping plant because combined with other crops. The manure and fertilizer using to cultivate this fennel, it is needed 15 tons per hectare compost and 90 kilograms nitrogen per hectare, 40 kilograms phosphor per hectare, and 25 kilograms calcium per hectare. All the cultivation effort is in order to increase the fennel production of fennel or essential oil production. To get the increasing production both on the mass production of seed and also the essential oil, we have to arrange all the irrigation with control and plant protection. This is a plant and grow development from the seed. This is the juvenile or vegetative performance of this plant until this is the flowering plants producing the flower, and until the seed. It is need almost 6 until 7 months from the seedling to produce the seed in the Indonesia. Harvest and post-harvest handling. Time of harvesting this fennel depend on the clone and the cultivation areas. It is note that the umbel do not mature in the same time. So the picking have to take an interval from 10 to 12 days. Fennel seed takes 170 until 175 days to mature from the seedling. This depend on the area also. Harvesting is done before the fruits are fully ripe and completed mature.
Ini aja ya, apa?
Seed of fennel have it could be harvest until the colors of the seed slight greenish yellow with the highest volatile oil content. This refer to the previous research. And umbel harvested are dried for 1 until 2 days and then in set for 8 until 10 days. It is not recommended to dry the seed in the oven more than 40 degrees Celsius because will decrease the volatile oil content. And also it is not recommended to offer exposure to the sun because will change the color of the seed and it is will reduce the quality. And avoid picking the seed after over ripening because it will lose in the land. It's easily to put off from the seeds, from the fruits. Refer to the phytochemicals research of fennel. Fennel seeds are used as spices and condiment. Besides contain chemical compounds, it also contain the essential oil. Fennel food contain volatile oils such as anethol, phencon, and pinene, but also has non-volatile compounds such as alkaloid, carbohydrates or nutritious values, phytosteroid, phenol, tannin, coumarin, and flavonoid. There are so many research have been conducted to evaluate the phytochemistry of fennel seed, as well as the all part of the Funiculum vulgare plants. This is the example of the essential oil compound contained in the fennel seed, such as transanethol, fencon, estragol, or methylcavicol, and alpha-pilandrin. And the essential oil of Adas is one of the main essential oil which are used in the medication formulas. It is a phenol compound, for instance, quercetin and caveolol. This is a glycoflavonoid contained in the seed of phenol, such as acatin, camphorol, naringetin, and isoharmestin. Come to the pharmacological aspect of phenol, we start from the ethnobotanical of phenol in the first initiated uses of this plant as medication. From several publications, it is revealed that phenol in the ancient times using to protect the houses from fire or accident and evil. This is such as plant protection, and also as an antidote for poisons, herb, or mushroom. In the ancient fertility rites for serpent or other venomous bite, to cover cold stone and earth floor, and so on and so on. So many traditional properties have been reported of the fennel seeds in the ancient time. The other use added to medicine to improve blood flow. This is very famous in Indonesia. Others using for spice for several cuisine also, such as gulai, kari, and rendang. This is used others as a food supplement during calving, grown in pasture to increase milk in herd. So it is also used to increase the breast milk also in Indonesia for lactagogum, scenting soap and perfumers, and to get rid of fleas. Seed are used for input and liqueurs also. Based on the ethnopharmacological uses of the fennel seed, so pharmacology research of fennel reveal that so many medicine or pharmacological activity have been evaluated, such as for antibacterial, anti-inflammatory, antispasmodic, for carminative, diuretic, antiviral, anti-stress, antithrombotic, and so on. So many pharmacological evaluation of the seed or seed extract. Beside the seed or seed extract, it is also have been evaluated the chemical content within the seeds, such as dianethol or photoanethol. The seed essential oil have been evaluated for oestrogenic, beta-myrcene or limonene from the seed essential oil also have been evaluated for hepatoprotective, transanethol for antithrombotic, 5-methoxysporalen for human liver cytochrome, and several kinds of chemical compound also evaluate for anti-radical scavenging or antioxidant. This is yeah, thank you. Yes, and the safety. Based on the research evaluation of the safety, it is revealed that this herbs that this herbal or fennel declared as safe herbal for human, and this is can use for any medication in human. So the acute oral LD50 for essential oil also shown that the LD50 is categorized as a safe ingredient. But notice for the estragol or methylcavicol compound from this seed, it is one of the main component of phenol essential, have been reported to have associated with the development of malignant tumors in rodent, but this is still not clear. It is still needed to be explored further by other research. But the potential of estragol to induce carcinogenesis still remain unclear. The economic aspect of this phenol that show that Indonesia is not belong to the 10 top of global exported in the world, because the exported country dominated by China, Vietnam, India, Egypt, Afghanistan, Germany, with a total export value more than $60 million. So this is the global prices also still not stable. This is the most highest prices of the phenol come from South Africa, or Cape Town, from Brazil. This is reached to $7.55 per kilograms. And this is the main producer of phenol in the world, dominated by India and China, actually, and South Africa. This is open to Indonesia to contribute more in the future to increase our production, so we can share our phenol into the world market. The conclusion of my talk today, I could say that Funiculum vulgare has shown an important medicinal plant use in wide range of ethnomedical treatments. Studies carried out in the past and present indicate that the phenol possesses diverse health benefits. This plant has been in use in long period of time without any documented serious adverse effect, so it is safe. Phenol has a wide market opportunity and has great economic prospect. And there are many areas of research related to this plant to be export, to be evaluated in the near future. And let me take this opportunity, because this is also another task from the National Working Group of Medicinal Plant and Traditional Medicine, for me to provide the information regarding the main topic for today's seminar is about the down insulin or insulin leaves, because there are so many doubts about the insulin leaf, since the insulin leaves in Indonesia provide by several kinds of species medicinal plants, such as Titonia diversifolia, Stevia rebaudiana, Smalanthus sonsifolia, and down Africa, or Furneria. Yes, and this is at a glance I will show all of you the habitus or the morphological appearance of Smalanthus sonsifolia differ with the Titonia diversifolia. Indicate the different size of flowers, different morphological appearance of the leaves, and also the critical differences between Titonia diversifolia and Smalanthus sonsifolia, which is that the Titonia diversifolia did not produce the bulb or tuber. That's all my presentation today. Thank you again for your kind attention. the time will I will return the time to the moderator. Thank you. So sorry, I cannot hear your voice, Mr. Moderator.
Professor Kerry and Professor Yuri for the very nice talk about forecast, and then Professor Kerry discussing about the stevia tea, and then Professor Yuli discussing about the ADAS or fennel, Phenylculum vulgare. Okay, now I open discussion, but before
Your voice, Prof. Sutopo, it's not clear here. Your voice. It's same. Prof. Kerry, how are you, Prof. Kerry?
Fine, Pak Asep. Prof. Asep, apa kabar?
Kabar baik, Prof. Alhamdulillah farmasinya udah jalan.
Oh, alhamdulillah. Selamat ya, Prof. Selamat
Ya,
juga
makasih.
untuk gelar profesornya.
Ya,
Satu.
makasih.
Hello, thank you, Professor Asep, Professor Kerry, and Professor Yuli. There are some questions I have concluded actually before. Now, before I have question to all of you, but I'll read the question all directed to Professor Kerry. Professor Kerry, the first question from Ibu Rabiatul Adawiyah. According to her, is there any contradictive metabolism between teh dia and nutrition metabolism, especially micronutrition? That's the first question, Professor Kerry. The second question from Dr. Paul Timothy Wu. Did you ever compare stevia to sugar from corn, such as product of Tropicana Slim for diabetic? And then the third question, from Vina. As we know, there are other sweetener contained in stevia rebaudiana, such as rebaudicide. However, why stevicide became the most popular compound in many diabetes research? Second question, how to minimize a bitter aftertaste of stevia leaf? Thank you, Professor Kerry. Okay, Professor Kerry, could you please answer to those questions first?
Thank you, Prof. Tata. The first from Ibu Adawiyah, the contradictive Tehdia with micronutrients. Yeah. Since the tea and stevia is beverage, so I think there is no contradictive. And then I search also the contradictive between the compound in Tehdia and macronutrient. Until now, I didn't find some publication.
Halo,
Halo?
Profesor Kerry.
Ya, saya sudah menjawab, sudah terdengar? Do you hear
We
me?
cannot hear your voice.
Ya,
Oh
saya
ya,
bisa mendengar. Prof. Sutopo yang nggak bisa dengar.
Profesor
Guys.
Kerry.
ya. Kalau semua bisa mendengar, Prof. Toto yang nggak bisa dengar kayaknya.
Prof. Sutopo, saya dengar.
Halo,
Prof.
tidak
Yuli
dengar,
dengar?
Prof? Yang lain mendengar nggak?
Dengar,
Profesor
saya
Kerry,
dengar.
we
Ya.
cannot
Berarti
listen to your voice, please.
nggak Prof. Topo yang nggak bisa dengar, Prof.
Iya.
Prof. Toto nggak bisa dengar kayaknya, itu ada masalah di itu kali. Di apa di kalau saya tidak unmute kok. Tidak di mute, sudah unmute. Mungkin panitia bisa bantu itu di sana?
Prof. Kerry lanjut aja, Prof. Karena kayaknya audiens bisa dengar deh.
Audiens bisa dengar ya?
Ya.
Baik, saya lanjut aja ya. Prof. Toto izin saya lanjut ya.
Soalnya waktunya.
Oh, baik ya, saya lanjut aja ya. Baik. And the second question is how about the comparison between sugar and a sweetener from sugar and sweetener from stevia? As we know that there is some sweetener also from stevia as a sweetener itself, like stevia sweetener, gitu ya. But sweetener from stevia, like in the market, other brand, it means the sweetener is the sweetener color is white. It's mean that already bleaching, already already the chlorophyll is already removed. Yeah. And then they are just to isolate the steviocyte. And in my last research, the steviocyte itself, it is not too good as the sweetener because there is some obstacle that we must to observe furthermore. So it is good if we use stevia by nature. Stevia by nature, I mean extract, not just single compound. Yeah. How about the sugar? If we compare the nutrition fact from sugar, from the corn sugar with the product of the stevia leaves, the corn sugar is still have carbohydrate nutrition in the present, although in the little bit presentation, but in stevia tea, we already can remove the carbohydrate, and then also the carbohydrate and the sugar in the zero concentration. This is from the sugar corn and the comparison between the sugar corn and the stevia. And the other is why we just take a focus on the stevia side, not riboside side. In molecular docking, we find that from molecular docking, we find that in the molecular dynamic, the stevia side is has a high performer to bonding with the receptor of stevia, receptor of PPAR gamma. So I think the tightening of the bonding also as a major major focus of us to although in the extract is they are some compound like riboside and then also stevioside, but the bonding is more strength in stevioside, and also stevioside is the major compound of the stevia leaf. And the other question is how to minimize the bitter. There is our there is our patent to minimize the bitter. There is some pharmaceutical technique to minimize the bitter taste or aftertaste from the stevia leaf. So in the Tehdia, the bitter taste of the stevia leaf is we can minimize, and then we have the good taste of the stevia with tea itself. Thank you.
Thank you, Professor Kerry, for the nice answer to the question. But there are some questions still. The first one, still to Professor Kerry, it is from Bapak Dadang Supriyatna, Balai Besar Industri Agro, Kementerian Bogor. Professor Kerry, relating to the healthy benefit, did you also study the effect of heat to the active compound of Tehdia, relating to heat resistant or not? Because we consume it by pouring to the hot water. That one is Pak Dadang.
Yeah, can you repeat, because I cannot hear.
Oh, okay, sorry. From Pak Dadang, can you hear Professor Kerry?
Yeah.
Okay, Pak Dadang is from Balai Besar Industri Agro Kementerian Bogor. Relating to the healthy benefit, did you also study the effect of heat to the active compound of Tehdia, relating to the heat resistant or not? Because we consume it by pouring the hot water. Thank you.
Yeah.
And then the second question is from myself, because no other question here. I have a question to all the speakers as my main task as a moderator. Okay, to Professor Asep first. Professor Asep, you have isolated some compound from the jatropha. Very difficult the name. Sorry. Jatropha. How will you later on in the future use it as a commercial drug? And do you think how long it will be? That one is for Professor Asep. The second to Professor Kerry. Professor Kerry, regarding the stevia tea, congratulations, you have got two products, and hope it will be successful in the market, either Indonesia and overseas. My question is, have you ever tried to isolate the active compound for the diabetes? If yes, what sort of compound you have found? Then as for Professor Kerry, and then the next question is for Professor Yuli. Professor Yuli, ADAS, because I'm also Javanese, I'm from Java, yeah. Because so that way my name is Sutopo. Content O is always Java actually. Okay, Professor Yuli, based on the active compound that had been isolated from ADAS Pulau Waras, is there any specific compound that you have applied, for example, as antibacterial or anti-cancer or whatever, something like that? Is there any specific compound? I mean, for example, this compound for this one, something like that. So there are another question. Maybe the first question I'll give to Professor Kerry, because you got the question, and then after that Professor Asep, and then Professor Yuli, please.
Okay. Thank you, Professor Toto. The first is a question from the Pak Dadang Supriyatna about the heating during the pouring the tea. Yeah, I already has a research for for the evaluating the glucose lowering or the hypoglycemic agent of the tea stevia or Tehdia before they give to the animal models. We pour the tea with the heating water, with the heat water, and then we also For the surfing manuals, also we are also give the information, use the heat water, and then the heat water, after the heat water we give to the to the animal models after the water is become warming or more cool. I think it is no evidence rise up about the disturbance about the tea after the heat water, because any kind of the tea that we serve with the teh dia is use heat water, and then also they show the benefit. And then the other is from Prof. Toto. Maybe Pak Dadang, if you want to ask more, you can also interact with me. And then Pak Dadang, the one compound that is isolated is a steviocyte as a marker for our product that submit to BPOM for the registration data. We use steviocyte. Why? Because in global research, they also have the focus of the steviocyte for the anti-diabetes. So also because teh dia, it's mean teh for diabetes or tea for the diabetes. So also we are focused on the steviocyte. But others compound, I believe they also have the other activity, because in our systematic review about the stevia leaf activity, there is just not for the diabetes, but also for other activity like anti-cancer and so on, as I already presented in my presentation. Thank you so much.
Thank you. Professor Asep, for you, please.
Okay, thank you for the question. That I have explained about my research or our research. I have done the activity of the Jatropha curcas and Curcosun B. We have done in the laboratory in Darmais Hospital in Jakarta. I calculate that is more than 10 years until now. We still check the activity with the other cells line. And then how long we can get the one compound from the natural use as a drugs. That I think this is it's a long time. Yeah? Maybe this is more than 10 years, depend on the budget, because this is very high cost for the research for the drugs. In laboratorium or in a journal, that activity of jatropon and curcosun B, this is better than the sorofanib and the other drugs for the cancer. What's the next? The next we will do about the clinical trial. This is we know about the 4 stages, yeah, or 4 steps. The first one step, maybe we need for 20 subject health, and then stage 2 we use more or less about 200 people for the use the drugs, and then step 3 maybe 800 until 10 people for use the drugs, and after that we go to the post-marketing surveillance. This is, I think, is very high cost for this research for clinical trial, and then we looking for maybe super from the factory or maybe for the university to get the grant, maybe the grant for the clinical this clinical trial. And I think this is a very long time. Yeah. I hope this candidate for the cancer, anti-cancer, I think this is better. Okay, thank you, Prof. Stopo.
Thank you, Prof. Asep. Hopefully it will be real in the next short time. Now, Professor Yuli, there is another question, Professor Yuli. Just I got it from the chat from Bapak Usman Siswanto. We may easily find fennel is sold as vegetable. Is it recommended to consume it in the long period? Thank you, Professor Yuli. So there are two questions. Thank you.
Okay, thank you for your good question, Prof. Topo. Yes, actually, I've been referred in my previous presentation to the pharmacological evaluation of phenol seed phytochemicals such as, for instance, dianethol and photoanethol.
Trouble. Maybe internet is not good.
Research also transanethol. It is so that very strong to have the anti-cancer activity by modulating the cell cycle arrest and other phytochemicals from fennel seeds have been evaluated for many kinds of pharmacological activity. For Pak Usman question, yes, in Indonesia, daun adas or fennel leaves are very popular in several areas, used as a vegetable and as a vegetable because the consume of the leaves are not so much here. I think it is recommended to be as a food consume every day. It is okay for consuming every day. Yes, thank you.
Thank you, Professor Asep, Professor Kiri, and Professor Yuli. Because now time is almost close to 1 p.m. and we have to have a break for a while, and then we meet again later on at 13:30. Now let us give a very lively applause to the three speakers. Thank you once again. Sorry, Professor Asep, Professor Yuli. The certificate and the other thing will be sent directly to you later on because we need time to do it. Thank you. And then I'm sorry for any mistake during moderating this session. Assalamualaikum warahmatullahi wabarakatuh. And I give back to Master Silakan.
Terima kasih, Prof. Topo, Prof. Keri.
Thank you.
Terima kasih.
Prof. Asep,
Terima kasih. Waalaikumsalam.
Ibu Yuli, waalaikumsalam.
Ya, waalaikumsalam.
Saya izin live ya.
Yes, ma'am. Thank you, ma'am. Thank you very much, Professor Sutopo Hadi, for guiding us in the second section. And also we want to thanks to all of the plenary speakers. All the certificate and souvenir from us will be sent directly to your home address.
Terima kasih banyak Bapak Profesor Sutopo Hadi yang telah memandu kami di sesi kedua. Dan terima kasih kepada seluruh pembicara pleno. Semua sertifikat dan souvenir dari kami akan dikirim secara langsung ke alamat rumah masing-masing.
So this is the end of the plenary session. Next, we are heading to the parallel session for oral and poster, which will be started at 1:30 p.m. All the participants may enter the room based on the instruction that have sent in the email after the lunch break. For the time being, PlayStation, we are presenting herbal and functional products of our college. Here they are.
Dan ini merupakan akhir dari sesi pleno pada hari ini. Selanjutnya, kita menuju sesi paralel untuk oral dan poster yang akan dimulai pada pukul 13.30. Kepada seluruh peserta dapat memasuki ruangan berdasarkan ketentuan yang telah dikirim di email masing-masing. Kepada para hadirin, harap menunggu. Kami akan menghadirkan produk herbal dan fungsional dari rekan-rekan kami. Berikut adalah videonya.